right, I think we're going to get started. It's 8:45. So good morning, everyone. Welcome to ARM's first workshop in 2024 on advanced manufacturing and cell and gene therapies. My name is Josephine Lembong. I'm the Senior Manager of Science and Industry Affairs at ARM. It's great to welcome everyone here, both in person and virtually. And we'll get started. We got a great agenda for you. So I think most of you here are probably familiar with ARM. If not, ARM is the global voice of the cell and gene therapy sector. We are a nonprofit advocacy organization that represent over 400 members worldwide. Our members are the technology developers, therapeutic developers, patient organization, tool and service providers, as well as medical research and centers. We represent our members both in the US and Europe on bringing safe and effective therapies to the patients. And we do so by convening the sector, advancing the field through data and analytics. We engage the key stakeholders and enabling the development of advanced therapies, as well as modernizing healthcare systems. So in our effort to achieve these focus, we have identified several priority areas, and today's workshop on advanced manufacturing is one of them. So the goal for today is to look at the successes, the challenges, and the opportunities in developing and implementing advanced manufacturing technologies, look at the role of emerging technologies in this field, and address some of the limitations around capacity and infrastructure to help bring these therapies to the patients. So you'll see a lot of different topics across the agenda today on the different sessions through really interconnected topics. So what is the latest in scientific and technological advancements in the field, both in manufacturing and analytics automation, the different manufacturing models, so bringing the therapies closer to the patients, distributed and decentralized manufacturing. What are the regulatory considerations to implement these? As well as exploring different partnerships models to implement these new innovations. So we rewind a little bit to last year. 2023 was really a breakthrough year of approvals. We have a total of 8 therapies that were approved in the US and Europe, including the first gene editing therapy by Vertex Pharmaceuticals and CRISPR Therapeutics for sickle cell disease. So this slide just has a list of all the approved therapies across different modalities and different indications. The CRISPR gene editing therapy was approved over 10 years after the discovery of CRISPR. So it's really a great milestone for the field. Okay, so this slide, I think, is just a snapshot of our sector, what it looks like in 2023 in terms of the number of developers, clinical trials, as well as investment. There's over 2,500 developers globally, over 1,900 trials, and nearly $12 billion in investment. The majority of the activities happen in North America. So there's a lot of effort, a lot of time, a lot of money being put into bringing these therapies to the patients. And if we focus in the US in particular, if we look at the number of INDs at the FDA Office of Therapeutic Products, or formerly known as the OTAT, Office of Tissues and Advanced Therapies. So this is what the number of INDs look like between 1990 to 2021. So there's a big increase of the number of INDs in the past 5 to 7 years. And I acknowledge that there's the last data point in this plot is on 2021, but we believe that the trend will continue up to now. So again, I think more INDs will lead to more BLAs and more product approvals in the market. And some of these advancements can be attributed to the advancement in manufacturing. So this is what the focus of our workshop today will be. In 2024, so another exciting year. We're in the middle of May right now, and there are already 4 approved therapies across US and Europe. So including the first the first adoptive cell therapy for solid tumor for advanced melanoma by Iovance, and the CRISPR approval in Europe as well. There are 9 decisions pending for the rest of the year, in addition to 6 total submissions that are still possible this year, although a regulatory decision is unlikely to be received until 2025. We, for more details around what these decisions pending and whether the approved therapies are, I will refer you and encourage you to read our sector snapshot that has all the pipeline for all the pending approvals. All right, so from the manufacturing side, right, so we're thinking about how we can bring the cell and gene therapies towards more global access. So these are all snapshots from various snapshots from the CRB Horizon Life Science reports from the last 2 years. So the reports included the results from the survey to various developers and experts in the field around where they are in the development and implementation of a lot of the different, what I call emerging technologies in cell and gene therapies. So, they ask questions such as like, how would you lower the COGs for cell therapy products? And as you can see, the automation and advanced analytics comes in the top of the most impactful factors in lowering COGs for cell therapy products. And then development of cell therapy platform process. So I think more than 75 of the responders say that they either already have a platform or they are developing a platform to be implemented in the next 2 to 3 years. So this will really streamline development for each developer to not have to start at square one again every time they start a new IND. Developers also are exploring different partnerships with hospital and clinics to be able to help bring these therapies to the patients. Again, more than 75% of the responders said that they're considering or already implementing a partnership model with hospital or clinics, but also acknowledge that there's a big barrier to be able to implement that model. And last but not least, there's investment in AI in different stages of development, R&D development and manufacturing. And over 75% of the responders said that they are already implementing or will be implementing AI in the next 2 years. So I look at these as, the 5 bullet points in the middle are all the key themes that emerge from the survey and what we heard from our members as well. And those are the 5 topics that we want to dig deeper in the next day and a half. All right, so I want to take a step back and discuss some of our efforts in helping our members also get to that manufacturing advancement. So one of the topics in the previous slide was on platform technologies, or what we call building blocks, I think for a more general term for cell and gene therapies. So we conducted a new format of interaction with the FDA that we call the scientific exchange. This is really a small roundtable format of multi-stakeholder spanning developers, academic, and regulators to really dig deeper into some of the emerging topics in the field that where usually a lack of guidance, and we think of this as a more proactive way to influence some of the guidance that are needed in the field. So we've done 2 of these format of interaction. The first one was on potency assays in 2022. And last year we conducted one on what we call the building blocks. So the idea is to have developers come into the room and present their case studies around what a potential building block is in 3 different key technology areas. So we have about a dozen or so case studies that were proposed by the developers, either on AAV, LNPs, or iPSCs. And the framework for the discussion is they will present what the building block is and how is it characterized and how can it be referenced by another developer and what resource savings does it allow by using this building block. So again, I encourage you to check out the white paper that was just published actually just a couple of weeks ago. So it's now out, and the white paper really summarizes the discussion from the room, which is around the viability of these building blocks. So as the developers presented, they get a discussion from the other developers and also feedback from the FDA around the viability of the building blocks either to be used within their own development program or by another sponsor. And we also summarize a few potential pathways for either a consortium or an industry association like ARM that can help further development of these platforms. And of course, we comment on FDA draft guidance as well. So I'm sure many of you in this room and virtually are aware of the draft guidance that was released at the end of last year on Advanced Manufacturing Technology Designation Program. We submitted a comment. In general, ARM supports the designation program. We think that it will help enhance the manufacturing innovations, which eventually will reduce costs and standardization across the field. We expressed concerns around the limitations on being able to cross-reference CMC data within the BLA applications. So we think that this language in the guidance will hinder technology development and is actually contradictory to the language around the statutory goals. And therefore, we recommend to remove these limitations in the guidance and revisit the rules to be able to allow cross-referencing data within the designation in the BLA submission. So we also suggest for a few clarifications around how this designation program relates to other FDA pathways, also a definition of a model drug and risk assessment. All right, so with that, in the next day and a half, we are going to discuss mainly 3 key themes, and these are big themes, so there's a lot of topics within each of them. We're going to talk about advances in manufacturing automation, the principles and challenges of processes and analytics automation, closed system and parallel processing considerations. The second one is role of AI and machine learning in cell and gene therapy manufacturing. And tomorrow we're going to focus on decentralized manufacturing. So all the manufacturing analytics considerations, the quality program, comparability, and facility considerations as well. So how to engage with us both in person and virtually. If you're here in person, there are 2 microphones on the aisles. So I encourage everyone to get up and speak to the microphone, state your name and affiliation before asking the question. For our virtual attendees, you can use the Zoom Q&A and chat function. We'll be monitoring the Zoom the entire day. We'll share a survey as well after the event wraps up tomorrow, and we'd love to hear your feedback. And as all of our other workshops, we'll be publishing the summary of this event in a white paper as well. Let me pause there, see if there's any questions. Okay. So since we talked about AI, I naturally went into ChatGPT and asked to describe to me what a CGT manufacturing looks like, what the future of cell and gene therapy manufacturing looks like. So if you go into ChatGPT and ask exactly that prompt, describe to me what the future of cell and gene therapy manufacturing looks like, it will spit out this long text, which I encourage you to try yourself because it's very, very difficult to read here. But I want to highlight, I think, a few themes that I think is worth highlighting here. So number 1, advanced robotics and automation technologies will play a pivotal role in streamlining the manufacturing. 2, artificial intelligence and machine learning algorithms will be utilized to optimize manufacturing processes in real time. And then there's comments around advances in cell engineering and manufacturing that will in turn shorten turnaround times and reduce costs. And there's, I think number 5 is around 3D bioprinting and organoid technology will revolutionize the production of complex tissue constructs and organoids for regenerative medicine. And last but not least, of course, there's comments around regulatory agencies will continue to adapt the evolving landscape of CGT manufacturing by implementing innovative regulatory pathways and standards. And very happy to see that there's collaborative efforts between industry stakeholders, regulators, and academic institutions will drive these, which is what ARM is doing and why we have this workshop in the first place. So probably not surprising that all of these themes are what we're covering. I promise I did not build the agenda based on what ChatGPT told me to do, but it's encouraging and again, probably not surprising that it's, what we're hearing from the members that we want to hear in this workshop as well. All right, so with that, that is my last slide, and I want to welcome our first speaker, Dave Smith from BioCentriq, to the stage.
Perfect. Thanks, Josephine. Welcome, everyone. Great to see so many faces here and I believe so many people virtually as well. Clearly, have picked the right topic to engage this audience in. It's my privilege and thank you to Arm to speak to you first, pretty much to set the scene, hopefully, for the phenomenal schedule that we've got coming up. So when I put this title together, I was trying to be provocative, but probably to the wrong audience here for not if, but when. I think for this talk, it kind of take us on a roller coaster. I think human nature will say that we always push, we always strive to do better. And I think, I for sure will touch on a lot of those points, looking at the pitfalls maybe of automation, some of the bottlenecks and challenges, but also some of the successes that we've had. I think we've come a long way in a pretty short time. So hopefully we'll cover all those topics today. Firstly, nomenclature, one of my favorite topics. I don't know whether it's because I'm an ex-Brit that's moved to America, but one of the things that I like to start when I start talking about automation is what are we actually talking about? What does automation mean to people? And I think when we talk about process automation, most people actually jump to bullet point 2 there, task automation in my mind, and it's really looking at each individual unit operation. Something like cell washing, instead of pipetting fluid in and out and then manual centrifugation and things, can we automate that with some of the devices on the market? That's just one facet, and I see it in kind of these 4 buckets. So starting at the top, process automation, probably the hardest one. Really, for any kind of hardcore engineers in the audience who understand kind of feedback, feed forward strategies, but can we measure something and make a decision based off of that? And can we do that in an automated fashion, as opposed to the world we live in today, where pretty much every single thing gets elevated to a human to make a decision? So that's process automation. Task I touched on. Test is another fairly obvious one, I think, when we move over to the analytics. Can we automate that? A lot more in terms of inline would be useful to therefore push onto that process automation. But offline obviously has its advantages and benefits right now. But still, can we automate what happens in a QC lab? Can we start looking at systems in place to do that? And the last one, operational or factory automation. There's a lot of when we speak about digitization, when we start looking at LIMS systems, MES systems, electronics batch records, warehouse management systems, all of those great things. And for me, we only really get true automation and complete automation of cell therapy when we have all 4 of these buckets completed. And I think quite often we look and we focus on just an individual one, and we don't tackle all of them. I think when we tackle all of them, that's where the big wins and the successes are. If we just take 1 or 2 of these pieces, I don't think we get to the true benefit of automation of our processes. And this is why we're doing it. For me, it's consistency, efficiency, and accuracy is the key drivers for why we want to go to automation. Quality is always an interesting one. Quite often we see that maybe we have to take a knock in terms of a cell yield or a potency or something, but instead we're going to get maybe some robustness, some consistency. And so I think understanding what those levers are and what we're really striving for is important as we look through these lenses of why we're doing automation and what really is the benefit to us. Scalability is a huge one. I think it's, the point where I would say in the last few years, we haven't worried too much about it, but now we're starting to see scalability affecting patient access. We're seeing that there's drugs on the market. We're seeing there's a patient population that can get them, and we're struggling on the scalability front. And I think most people here will agree that automation can obviously really help with that. Again, it comes down to the consistency and the quality. The next one I touched on is cost of goods. And I think it's a really interesting one. You'll see I have it here as a driver of automation. I'll drive into the pitfalls later on and say it's actually a pitfall of automation as well. So I think it's a really interesting one to assess. And it's very easy to build a a case to say that I'm going to automate something, it's going to reduce the cost overall. But I think it comes back to that first slide I showed. I truly believe we can get the cost down significantly with all 4 of those levels of automation. If you're just doing one, you may actually start to see an increase in your cost of goods before anything starts coming down. And the last one, sustainability. There'll be a lot of talk and a lot of people sort of worried and concerned about workforce and that element of things, whilst we're still in a very manual process setting. So that's a big driver on the sustainability. I think I've put reducing supply chain risk. It may reduce it on a workforce development side of things, but it's going to increase it on maybe a consumable or an equipment side of things. So how do we balance that one? Which one's easier to control than the other? So as I mentioned, driving into the pitfalls said it would be a bit of a roller coaster. It's not all rainbows and sunshine out there. So these 4 areas, maybe we'll start at the bottom just to mix things up. I mentioned cost. For most of the processes where I've implemented automation, it's actually increased the cost of the therapy. It hasn't decreased it. I've taken a manual, low cost, cheap consumables, things like that. Now I've had to go and spend 100, 200K on CapEx for equipment. I've had to put 2,000, 4,000, 6,000 on a consumable for every single batch. It's actually increased my cost over time. It hasn't decreased it. In my mind, the reason, I've only done one of the things. If I've started to link all of those things together, and if I've looked out over time, one of the issues that I feel we struggle with, we're still at pretty low numbers of patients that we're treating. We're not getting, and I feel, the economies of scale. Now, it's not going to be anywhere great to some of the other industries. When we look at the autologous market, Allogeneic, sort of a different driver there, but there is still economies of scale as we drive into these things. Reduced flexibility. That's a big one. I think where we live today, automated processes definitely increase our consistency, but they do reduce the flexibility. If we want to increase the flexibility, they are very large, very time-consuming, very complicated instruments to keep that flexibility. So I think understanding, again, the driver of when is the flexibility needed? Maybe it's needed really early on as you're developing. As you get later on, you can afford to reduce your flexibility. So now we look at that automation path from discovery through to commercialization. When is the right time? What is the right time? What level of automation do we need as we go along that journey? Failure consequences, one that I feel a lot of the companies don't necessarily think about, and I think hopefully we'll get into it into the panel. What happens when an instrument fails? Okay, so now we need a backup one. We need 2 of them. If you're midway through a process, how do you convert over from one piece of equipment to another one? Can you do that? Is it easy? How are you going to do that? There's a lot of pitfalls that I see that a lot of us aren't planning for. We assume that everything's going to be great and nothing's going to fail, but the reality is that the equipment in the market today, it fails. And I'd say it fails quite often as well. A lot of things, were built from a research standpoint. They weren't necessarily built to be run 24/7, 365 days a week. So how do we deal with that? What do we do if there's a leak on a consumable set? If we've managed to, paint the theory that we're now in a CNC space and something's gone wrong, do we have to have a backup lab? Do we have to have an isolator? Do we just throw the thing out? How do we deal with that? What's the consequence? Sometimes automation can hide some of those. And the last one, the black box effect. I think there's a reason why in research, I would say nearly every single process I've seen has phenol red in it. As you go, everyone tries to remove phenol red. But people like to see, is it about right? Is it kind of the right color? People like that gut feeling. And sometimes you can lose that as you go through into automation. You put it in a black box. Some of the hardcore researchers just don't trust the automation because they can't see what's going on. They don't feel what's going on. And I'm not saying we should put phenol red in everything, but it's, as we think about automation and do that, how are we ensuring that we're getting that gut feeling? At the end of the day, it's going to be those researchers that are going to be driving the innovation and bringing those next products forward. We need them to feel confident in the automation to be able to adopt it as well. So moving on to the positivity. So I tried to put a timeline together of automation just to see how far we've really come. And I think if we go back into kind of the 80s when Rosenberg first managed to put TILs for cancer, And there really wasn't much going on in terms of automation. But what there was a blood banking industry that had put some money, some ideas and concepts in. So some of us may remember the COGs 2991. I'd say a workhorse back in the day, probably less used now. And so a lot of that industry was just, we were borrowing instruments from other industries, and largely the blood banking industry, as I mentioned, which was great for proof of concept. We really had to evolve on. And so that's when we get into kind of the late 90s, early 2000s, where we started to see specific cell and gene unit operations. And that was great. Suddenly there was a market for it. Suddenly there was a movement towards it that could really help us develop our processes. That really expanded over into the sort of the tens into the where we are today with, and I think it was Deloitte that coined it, and I loved it, this automation islands. And I think that really paints a great picture of the idea that it is a single unit op, you're doing one thing, it's not really connected to anything else going on. The only way it's connected is through human interaction, which we'll get onto. And so all of these technologies were great. And you'll see, obviously, a lot of them were task-based. And then we started moving into some of the test-based automation. So obviously, I remember, you know, the 3 different people in QC using the hematometer, getting 3 completely different answers. And as soon as that nuclear counter came out, you know, a big kind of movement over onto it, it simplified everything. Perfect example of sort of test automation and a success for that as well. We also started to see software, TrackCell, obviously, you know, one of the forefront runners on chain of identity, chain of custody, and even starting to see things like Autologous with an electronic batch record devoted to cell and gene therapy. I mean, that really was a great proof point to say there is an industry here, there is a need here that maybe is not being served by some of the larger kind of MES providers that maybe Big Pharma have used previously. We then move forward to maybe, let's say, where we are today, you know, end-to-end, maybe not exactly there, but definitely joining these islands of automation together. And I think you see that with the cell shuttle, and you see that with cellular origin system, and there's others, Multiply Labs, things like that, that have come out with these systems that now you can remove the human, join in all of these different separate unit ops together and replace that with a robot. Right? I think that's starting to now reduce the skilled workforce that's required, the amount of labor time and things, you know, really start driving to, you know, a great point where we need to go. And for me, the next step gets to kind of integrated facilities. I don't think we're there yet, but we're definitely well on our way to it. But I think if we look back all the way to kind of the 80s to where we are today, that's quite some journey and quite some technology that's been developed. You know, if you think when that first commercial cell therapy came through, there's a lot of work that was done before anything was even commercial. And so I think, you know, for me, automation has come a long way. I think 10, 15 years still at conferences, everyone complains that there's a lack of automation and we need to do more. That's the roller coaster I'll take you on next. But I think there's also a lot of great work that has been done. I wanted to touch on the adoption of automation. You know, I just mentioned that there's a lot of great technology out there, and sometimes it can be hard to adopt that automation and really see the drivers for it. You know, I think, you know, that first bullet point, high cost, high risk without adoption. You know, the cost of the equipment and consumable I touched on, the cost and the risk in development. You back a piece of technology, it doesn't work for you. How much have you invested to find out that there's a failure point there for it, or it doesn't quite fit your need? How do we overcome these challenges? Comparability, such a big question mark. How many runs do you have to do, depending on what stage you're at? What do I have to do? What do I look at? How do I do it? So many unknowns there that it scares people to even try and do it. And then time. Most people are running so fast, most groups are running so fast. Do they have time to look at new technology, to start thinking about adopting technology? I'm not saying it's the right thing, right? It's to look all the way at the end. What's your end goal? When are you going to do it? Plan everything up front. But it's definitely a factor with a lot of the clients that we see as a CDMO, that they just want to get into the clinic as fast as possible, and they want to use something off the shelf, be done, that's got a proof point. I mentioned the islands of automation. You know, they're very common. They're largely used throughout the industry right now. But if it's just a single point, does it really assist? If you're using a piece of equipment that your operator still has to sit in the clean room and watch, just in case it alarms or something goes wrong. What have you actually gained by using it? Maybe you've gained a little bit of, robustness. Maybe there's some scalability you can start to see if you've got multiple islands where, a couple of operators can look over everything. But if we're at single numbers, a single products in a clean room, it's not really assisting too much until you start building all the other levels of automation in as well. There's a big drive to wait for clinical proof points. There's a large burden on the front runners in order to bring a new piece of technology through into a clinical trial, check that it works, all of the development work. And I would say also to bring the technology provider along with you, right? There's a lot of lessons that have to be learned by both parties. Have you got the right testing done on all of those consumables for me to be able to use it in a clinical trial? What do I need to see? What does the FDA need to see? Have you got different files that are registered with the FDA that I can pull on things? There's a lot of work that goes into a new piece of technology going into a clinical trial and being used for humans. Technology not meeting the changing needs of the industry. As you said, how great we've done over time, but I do think that the industry is moving so fast. Right? A lot of the technology has been built maybe for a 14-day CAR-T process. Or if we're in a 3-day or a 24-hour CAR-T process, is that the same technology that we need? Are we going to be stuck 13 days in a bioreactor? Well, do we not even need a bioreactor now? And so I think understanding how rapidly the industry moves and how does technology keep up with that? It takes a long time to develop technology, to do all of the testing, to build a consumable, etc. They really kind of have to work together in order to understand what's coming next and where the technology really needs to go. A lack of standardization. Now, standardization on one hand, innovation on the other side. How do you balance those two? It's a real pondery of how we do that. But there are easy things that we can standardize. Tubing, for example, different connectors, API in terms of just getting the equipment to talk to one another. So we can start moving data between systems, making decisions. What came off of this system, now the next system knows exactly what the volume was or the cell count or whatever it may be. There's simple things that we can standardize that I don't think hampers innovation. And so thinking that one through is crucial. The last two questions I put down there, really driving, I think, to some of the great talks you're going to hear after me. How do we transition from discovery to GMP? I've mentioned it a few times now, smaller scale versions. We can't be at scale when we're doing discovery. This industry is far too expensive in terms of even just reagents, labor, things like that. We need to have small scale models that translate easily. We don't want to have to be doing comparability from discovery into phase 1 and then another comparability from 2 into 3. How do we get something where it's more of a fluid journey from discovery all the way through to how we're actually going to manufacture the products for the patients that need them? And then the last one, when we start looking at digitization, obviously there's a lot of great talks coming up on AI. We still need human decision makers throughout our process all the time until we can start removing that. That's when the cost of labor really starts to come down. I mentioned that we have to keep operators in the clean room in case something goes wrong. Some of them is just a visibility, what's going on, documenting what's going on to make sure we've got those proof points. Are we at a point where AI can start doing that? Can we start using ChatGPT, as Josephine did, to make decisions? Do we have enough data points? And I think there's something in terms of, if we take the entire industry, I believe we probably do. But when not only in the islands of automation, but the islands of companies as well, we've got to work together in order to make that feasible. So my last slide on this sort of future perspective of where I think needs to go, there is a ginormous patient need out there. We all know that. That's why we're here. How do we satisfy that? For me, is solving some of these challenges. The clean rooms, the manufacturing facilities are extremely expensive. Remove the people, remove the reagents, remove everything else, right? Just running a GMP clean room is expensive. The amount of monitoring you do, the amount of cleaning you do, every time you need to be doing plates, you need to be reading that, looking for organisms, things. There's just a huge expense when you're trying to run something at an ABC level. So how do we decrease that? How do we get into automated systems? How do we get rid of the dirtiest things in our clean rooms, which are the humans, lower those grades of those clean rooms, close the systems, start looking at continuous and parallel processing. That's where we're really going to start seeing kind of cost savings. If there's one product in a clean room with, 4 or 5 operators working on it, that's not great. If we can do 10 of that, divide that cost through by 10, there's a logarithmic saving already. And so starting to think about how we can do that. Reducing the workforce gap, I think, is an obvious one as we start moving into automation and how we can utilize automation to truly remove the labor. And I think it's I've given a lot of examples where we're using automation, but we're not actually reducing the labor. So we have to think through about how do we automate things to reduce the labor. There's a lot of things that are a lot easier than task automation. Something like flow cytometry. For every single patient where you may go into bioreactor once, you're probably doing flow cytometry 4, 5, 6 times. Already there's 6 times the number. So that's a huge one. Human errors. I touch a lot of humans as if I don't like humans, but removing them is a crucial one. Increasing our data banks is huge. And then the last one there, which I think is the biggest one for me, and we've seen even in the news over the last few days, is how do we partner across the industry to help this? I think there was great news with obviously the Galapagos came out with yesterday, I think it was, or this morning I saw it, with how they're partnering with Lee and his team up there. So I think more things like that where we can help each other. What is the island of knowledge that we have where we can help each other to move forward? And I do believe integrated facilities is the way that we have to go. Fully automated, all 4 of those levels of automation working hand in hand will get us to much lower costs of goods, not only for autologous, I think there's a big push on that, but also on the allogenic side of the market as well. And I think when we start looking at integrated, does that start to enable more of a decentralized mindset as well, which we're going to hear about later today as well. So thank you for your time. It's great to speak to all of you.
Yeah, we have time for a couple questions for Dave.
I'll start. Dave, thanks for the great talk, and thanks for throwing us the bone on the exciting news. We'll touch on this tomorrow as well, I guess. But as someone who sits in a contract manufacturing facility and business, how are you and your team thinking about this move toward the potential for decentralizing some manufacturing for some products, not to say that it's going to take over and put centralized manufacturing onto the business, but can you talk a little bit about how your team is thinking about playing in that?
Yeah, and I think it's, we'll go back to terminology about what we even mean by decentralized. I think there's a place for a lot of different cell therapies, I think. So right now at BioCentriq, the view is, sticking with the facilities that we have, not necessarily move into a decentralized model. We think that there's enough of a market out there for how we've built our systems, our infrastructure and things to not need to go to that. I think it's one, take a step back and get rid of my BioCentriq hat, that there is also a huge need and a huge benefit to be able to move in that direction. And I think it's important that we build the infrastructure as an industry to enable that. I think as we start looking at, the therapies now going, outpatient, for example, it makes a huge difference in terms of even the cost of administration and the effect they have therefore on kind of treating patients. But I do think it's something we need to look at. I think it's something that as an industry, we need to understand better, even what that means, right? I think a lot of terminology as well is what is point of care versus decentralized. To me, they're very different. But I know for some people, they say, well, no, they're exactly the same. And so I think understanding, is decentralized just many manufacturing sites around, or is it actually at the bedside? Right? I think they're 2 very different viewpoints. I think bedside we're a long way from, but when we start looking at 24-hour processes and things, I don't see how a 14-day process is going to go bedside. Now a 24-hour process, and now we've got the biology and the understanding of that. That's a huge enabler for how we moved in that direction.
Carl Coe, Arbor Biotechnologies. You mentioned one of the challenges like moving from early discovery to kind of like GMP scale. Arbor is at a point where we're very early on, very discovery. And you're right. We do things very fast and try to move as the clinic as fast as possible. The question I had was, does your company or other kind of companies in the similar vein create small scale processes that are geared for discovery or very early so that hopefully you can move very quickly? I know you guys focus on kind of like the GMP side, but is there kind of a need there to build kind of like a very early discovery style process or kind of like box that would work early on?
Yeah, I think there's a need. I think the hard bit is standardization. So we launched 9 months ago, I think, Leap. So that was our idea. We've got a standardized GMP ready to go CAR-T process. So we can just utilize that. The equipment set up, the clean room team is fully trained on it. So we can get tech transfer massively down because all the work's been done if you use that process. The problem that we see that everyone comes in and say, "Oh, I don't want to use that process. This is my process. It's much better than that one," or "This we do slightly differently." So now you're starting to change the process slightly. So now we're rewriting SOPs. We're having to redo the training. We're rewriting batch records and slowing it down. I think the same is going to be true at those small scale models. People are going to come in with their bespoke way of doing things, and they're going to want a small scale model for what they're doing. So I think, maybe provocatively, but I would say, does it matter? It doesn't matter which bioreactor I use today if I'm very early. Maybe it does if you've got a future plan. Does it matter how I select? Does it matter whether I use, Dynabeads versus Transact versus if you're just trying to get speed into a proof point, the answer is no. Now, if you're trying to build a therapy, you're looking at commercialization. Yes, you're going to get a very different product depending on which bioreactor you use, which reagents you use, and things like that. And so until, and I think different people have different ideas. Like some clients don't care about that. They want to go as fast as possible, get those proof points, and we'll worry about it later.
All right. Thank you, Dave. We have to move on to our next speaker. So thanks again, Dave. We're going to welcome Jason Dowd from Bayer for our next talk.
Good morning. I'd like to thank the ARM organizers for allowing me to speak today, and I'm looking forward to presenting our platform development at Bayer. For the slide. Okay. Oh, okay. But while we fill the space, today I will be speaking about our platform development, our end-to-end consensus platform that we're trying to build. I will focus in directly on our 2D robotic systems that we've been working with. And while I look for the laser pointer. Here we go. Just a second. Todd button. Forward arrow. Okay. Thank you. Sorry. All right. All right. So I'll briefly go through where our company profile and where we're working on, and into our industrialization approach and our advances in the cell therapy platforms. And so we work with our industrialization or in innovation partners, and they're over here and you'll recognize Blue Rock and AskBio. And this is what they do well. They do well on the research. They do well on formulating our initial products. But what Bayer does well is in the manufacturing and from the development and the manufacturing. So our needs overlap, but we certainly have the capabilities to look at how we develop platforms. And the goal is to get the patients in need. And so we really refer to this as an industrialization engine. And so this is for the first product is the bemdaneprosol, which is a Blue Rock asset, which has been going through phase 1 trials. And it involves a surgical suite. Burr holes are made on either side of the patient's head. And there's an insertion of essentially of almost a cell paste at that point because it's at such a large concentration. But it's meant to replace the dopamine producing neurons. And these have and there's been showing excellent results from the phase 1 trial. All the patients have demonstrated that they're getting better and better and better. And so we're actively enrolling patients later this year, or Blue Rock is, with the phase 2 manufacturing. The phase 2 manufacturing was completed at Blue Rock in Toronto, whereas the phase 3 manufacturing is actively undergoing in Bayer and Berkeley. And so this is our industrialization approach. And I could go into lots of different topics, but I think I'm mainly going to talk about the people and how we got there, and then some of the facilities, and I'll highlight those as we progress. And so why I joined Bayer was that it has a tremendous engineering history, especially in the German facilities. And we do have this AIMS group. There's around 70 FTEs, and there's very good capabilities in terms of understanding what's happening in bioreactors. I won't be talking about the 3D bioreactors, but I guess my point is watch this space, because we've had some excellent results on both the ESC and iPSC cell starting materials. But the point is that with this tremendous engineering capability, we've been able to build the prototypes, of which I'll speak. And the focus is on the 2D platform system, just namely because the 2D system came to us from Blue Rock, and that's what we were seeking to industrialize. These are the brand new CGT's manufacturing space in Berkeley, $300 million facility, plenty of space to do plenty of things. We've occupied the first floor, or the top floor actually, for the Parkinson's application, but we can also build out the following things. And we have the nice thing is that we have a 30-year lease with the city of Berkeley to basically replicate these kinds of spaces, and with a potential of up to 5 more. So the goal here is a long-term manufacturing capability in Berkeley. So this is really the starting point, the what, and you could probably lots of folks can generate this kinds of slides in terms of what we're looking for. We are looking for modular and flexible, end-to-end, small footprint, and many of the things that Dave talked about just now. And we are looking for it to be highly integrated and automated as we progress, with the goal that we accelerate the asset development time to market and really get to the competitive cost of goods and in the allogeneic framework. But and so this is a slightly older slide. My legal didn't allow me to update this too much, but the goal is to have this flexible cell therapy platform that goes end to end. And when we say consensus, meaning that we hope that maybe these islands of technologies, but also the islands of companies. I liked David, so I'm stealing it. Those components there, and you can start with various cell types. You might have some small scale, but then you're going to have some options on where as you progress to the various stages of the process, and even as you get to patient dose administration, because one of the parts that we're literally looking at is how do we administer these products, and how do you automate some of that production. And I'll only be focusing today on the 2D system and some of the work that we've been doing for the last 3 years. So this is more of a philosophical slide. It'll build as we progress. And so clearly we have the operators struggling with their manual systems. And there's some variability operator to operator. Never mind the biological variability of the starting materials, but how do they manage these things? And we do have a generation 0, which I'll show some pictures of. And we are going to be looking at maybe a 0.1. It'll be a question mark whether or not we go beyond that, frankly, because that'll be what we'll be using for the phase 3 manufacturing. But ultimately, what we'd like to bridge to post-launch is the generation 1. And this certainly does have some components of where you will be looking at different iterations. But I did want to, again, define the generations which is largely equipment-based, and then the subsequent iterations and the adding of analytics often is what we're going to be looking at as we progress through from manual to automated. And here is a fairly simplistic kind of breakdown of where to go. Okay, this is kind of a 2x2 square, and I think this highlights some of the. It's another perspective on what David was talking about. Because if you have very little robot or automation focus and you don't really focus on the biology, it's very, very limited value of what you're doing in terms of automation. If you focus too much on the robot and not enough on the biology, then you might suffer with getting too specific an application where you're not, it's only good for one type of cell, one type of media, and then if you change the biology, it's not applicable. If you chase biology too long, you're trying to solve too many problems, and you might never get there. So there is this kind of balance between the biology focus and the robotic and automation focus, which I feel is important. And so this is ultimately, looks at what the value proposition is, and these arrows, in terms of higher quality, process understanding, yield, and FTE costs, and they all have different starting points and end points. And if in a longer discussion, I'd be happy to rationalize where these go, because based on some of our experiences, because you might not necessarily lower FTE cost initially as you're moving from manual to as you progress through these different stages, but you do expect that there will be some improvements over time. And then finally, there is the cost and effort that goes into these productions. All right, so you can see a little animation here. This is our generation 0, which is our launch capable platform that we're, as I mentioned, producing the phase 3. We have it's an off-the-shelf components, the cell manipulator, but we did do some bespoke integration with liquid handling, and so the pump and the tubing sets and all the rest. And so, we can handle 3 CF40s or 6 CF10s, as you can see in this setting. And, we're also looking at how, and I mentioned the additional iterations. So there's going to likely be some modifications to the carts, because you have to roll these carts in and out, and various analytics that we do want to add in order to improve the process understanding. But this is the story of what we're calling Act Smart, which is automated cell therapy using smart robotics. And so the goal here is that you might start in Boston or Toronto, where Blue Rock is, and they have their best operator, doing traditional manual processing, how to slap a flask, and how do you move those in order to detach those from the cell. And so what this, you can get 4 sensors. This particular example is from, how big they were when Apollo went up to the moon, but obviously now you can get them almost the size of, almost key card size. And with that, then there's a translation so that the robot can hear it and or can understand it, and then the robot starts mimicking, and you can see this up here. You see the robot start mimicking what the operator is doing, and so that involves the consistency, with the goal then that can be then transferred to the Berkeley side with one of these robots, and then and then progress those. So here's the advanced prototype that we have, and it'll show some videos here in terms of what the robot arm does. It's in quite a confined space, again, using off-the-shelf incubators in order to try to reduce the cost. We didn't want to make something that's too big. The goal here is also how you translate those manual motions. The robot's quite capable of doing that very consistently. But we also wanted to start integrating the analytics. And so we do have an online confluency estimation. And so what you're seeing here is actually the entire bottom plate of a cell stack, say it's a CS10 or CF10. And so you actually get 5 by 7 photos similar to this, and every 6 hours you see the estimation, and you can do some simple statistics to see how how the confluency is increasing over time. And so admittedly, this is only the bottom layer. We will be looking at how we increase that automated real-time monitoring for the remaining layers. This bottom panel is really looking at how the quality of the imaging, because you can get scratches and bubbles and other things. And so there's a lot of fault analysis and detection that needs to happen in order to do that. And so an operator could go into each point here and see what's happening with at that particular point if there's some point of question. So this is the kind of automation that we're looking at. Here's a simple schematic. Like we did have the goals to monitor and control not only the bottom layer but all the layers, and we wanted to estimate the confluency and really kind of get so that this could be put into an AI and an automated system. The challenge, of course, is that it's very hard to visualize those upper layers. And so we're looking at how various analytics. And so in the green, this is what we've done. And what I've showed you is we do have the classification problem. We have got a system, and there's a publication in preparation to show this. Well, we're also working with partners, and perhaps there's people in the audience today that have some interesting ideas, and I'm happy to talk about that. In terms of looking at alternates to the metabolic route, because you can certainly analyze for the metabolites like glucose and lactate, and in order to predict what's happening in all the different layers. But we do have some approaches here. I can't speak about it yet, but we will soon have something. The goal here is that Bayer will complete the development and quantify and qualify the performance in GMP. And so this is our roadmap. We're also seeing that imaging is highly powerful. And so this is an example of using in flow. There's some various cameras that can operate to assess the cell concentration, because David mentioned the nuclear counter, and certainly that's been a real step forward. But even in depending on the biology, whether it's an ESC or an iPSC, sometimes you do get biases, and our team has seen that. So we're looking very extensively at imaging around the circle, as it were, and both for the adherent and for the suspension sides, because we see some real value in also that look and feel and a component. And so yeah, that's certainly where we're headed. In terms of where we are going for the digitization, certainly we are creating the data lakes and trying to collect the information from, again, I'm going to steal David's equipment islands, and trying to create these into a suitable data lake that can be cut up, and so that we can do the act and we can automate and visualize the analytics. I do have my colleague here from Bayer, Oliver, if you could put your hand up if anybody has any more detailed questions about what we're doing with AI. Oliver leads those efforts at Bayer, and so he'd be happy to have a good discussion about that. Yeah, so this is my concluding slide. In summary, we've progressing on our end-to-end industrialization platform. We're not done yet. You can see that there's some further generations that have to happen. And with typically in a platform, you tend to focus on the bigger problems, and you get very good at those particular problems. I would say we haven't been as good on the filling side and the patient dose administration side. Those automation processes have to progress significantly, and that's what we'll be focusing on for the next 3 to 5 years as we try to build out these allogeneic therapies. The goal with allogeneic therapies is to capitalize on those cost of goods savings as you go up the exponential or the logarithmic size of therapies. And with what we have accomplished with the ActSmart system, I'm sorry the text is a little small here, but we do think that we can produce about 50 billion cells per batch even from that small scale system. There are multiple systems that we're looking at that will build off of that. So it's really a question of scale of what we will do, what will we put into the Berkeley facilities, but we do think that we can produce in the tens of thousands to hundreds of thousands. And certainly that's the aspiration for Parkinson's disease. As we know, there's millions of patients worldwide that get this disease. We do want to provide suitable therapy for them. So I think I'm a little bit early. One thing that I will, if you'll forgive me, I will go back to this slide. And, I know that I painted the picture of this kind of going one way. But what the nice thing is that, we can also go this way as well. So if there is an operator that, that suddenly is the best, we feel that they could be standing outside of these units and actually tapping and clapping these flasks in order to assess the performance. And so that speaks to that look and feel. We do feel that you need that biology. Variations will happen with media components, with cell lines, and we need the robots to be flexible enough so that we can program them to do what we want to do, because one size does not certainly fit all. We certainly, as a quick anecdote, we did this first work with iPSCs, and it worked beautifully, and no challenges with getting to quite large scales. But with ESCs, different media, different cell line, where we had some challenges. And so there's a constant balance between the biology and the automation that we need to progress. And thank you all for your attention. And for that, now I can take some questions.
So thank you very much for that presentation. My name is Bob Lekich from Global OTIT. Something that's been rolling through my head, working in the space for a few years now, and automation has always been looked at, from a lot of different perspectives, management as well as the engineers, try to automate something to make it faster, to make it more efficient, to reduce quality issues, what have you. But what I saw in your slides, and I really think it's important to bring up, we automate to create knowledge.
Absolutely.
I think that's a fundamental issue for this space here, that we need to get out of the mode of waiting for automation until we have a commercial product. We need to automate so we learn earlier. So from a QBD standpoint, from a process understanding standpoint, we are evolving and aligning with our knowledge that we're creating. And so I just want to throw that out there.
Yeah, no, that AI and machine learning approach, we're questioning whether or not it will truly close the loop. But if it can predict when you should harvest that batch, instead of 9 a.m. because the batch record says so, okay, maybe it's 12, maybe it's 3 p.m. And the goal there is so that you get the right quality and the consistency is there. But because the yield on the cells is the key parameter. The cells are the product. It's not the amount of protein. And so if you're just dealing with protein and traditional biotherapeutics, okay, you could probably harvest in 9 o'clock, and it's okay, you're going to have some variation in your protein, big deal. But in terms of being able to predict and tell that operator and tell that supervisor, hey, be ready, because this is what's happened, because the variation on the inputs leads to a tremendous variation on the outputs.
Right.
And so we need to have those signposts along the way so that you can predict whether that trajectory, that cell journey is absolute.
And I think, in this space with cell and gene therapy companies, who are, a lot of them are startups or very early in either product design, if you will, and the clinical phases, to be able to learn more quickly with the infrastructure of the data analytics, the data infrastructure for having a data warehouse or data lakes. All these technologies may seem very down the road for a lot of companies, and I challenge that every day, right? We cannot wait to do that downstream. We need to do that upstream. And I know that's a challenge because funding is always an issue, but without learning and without creating new knowledge that will help us both in the short term, medium, and long term, I think that's a change in culture. That's a change in the way we do business. And I think That to me, I think is really important. I don't know what you
Right.
think.
No, I feel like I should be slipping you $20 now, because, you know, this is where we need to go, because that's also part of the piece with Act Smart, is that it could handle a T flask, or it can handle a CS10 and a CF40. So I have my built-in small scale. I know that I have all the operations handled the same way, you know, how the flasks are handled. So it leads beautifully into process characterization, because that is really where the industry is a challenge. Like, how do you characterize a manual process that you've taken it out of an incubator and leave it for a variable length of time? Like, how do you validate that operator? I want to get it so that you could do it at small scale. You could keep it in T flasks if you need to, or you could scale it up. And
This is the same challenge that I saw personally working in biotech organizations like Merck, where we were T flasks and Erlenmeyer flasks with shakers, and people challenged to say, can we really do this in a bioreactor? And, you know, it was the same issue, that the fundamental concern that we don't know how to do that and learn from that, you know, and eventually we did it, right? It just took a really long time. And what I think you're showing here, at least in my opinion, is that it can be done. It's just that we've got to be strong enough to create a new way of how the organization looks at technology. It's not for technology's sake. Never, right? We always think about it from a business perspective, but at the same time, the business is to learn about how the process really works and what makes it work correctly. You know?
absolutely. Yeah, no, it's about improving the knowledge of their systems.
I'm sorry, we have to move on to the next speaker. Jason will be in the. We have opportunity in the panel session later as well that Jason will participate.
Good morning, everybody. My name is Stefan Braam, founder and CTO of Cellistic. We'll continue to talk about iPSC, but more on the upfront side of the process. So creating gene-edited master cell banks, and how we are implementing automation to do that, to solve the fundamental issues, how to do this in the right way in the shortest time frame possible. So we are an iPSC CDMO, fully focused on building these manufacturing processes, and we partner with therapeutic developers to make those processes available to them. So fundamentally, what we realized especially in the immune space, where we are focused on, that we see very, very good autologous treatments that are not scalable. The manufacturability is not really there. The quality and characterization is so. I think what we are seeing now is that we want to see precise genome integration and not random integrations into the genome, which is very difficult in an autologous fashion. A price point that is high, and a price point that will not go down because of scaling, because that scaling effect we don't see. And so repeat dosing is not really possible. So essentially, this is a process, and that process will be very hard to make cheaper and make accessible for patients. So what we're focused on is to turn this to a product. And so the way to build a product would be to make it allogeneic, make a master cell bank, and manufacture from that master cell bank in bioreactors, essentially like we have seen in biologic manufacturing. So there is a very big parallel to biologics manufacturing here. So once you do that right, you actually get manufacturability, you can control quality in the right way, you can characterize those cells in the right way. And with that, price should go down when economies of scale start to emerge into the manufacturing process. This is exactly what we do at Cellistic. So to the previous discussion, we are actually focused on CMC from day zero and purely focused on building these manufacturing processes in such a way that they are scalable from the onset, because iPSC biology is actually very complex. It is actually a very big risk if researchers are playing in 96 well plates and in 6 well plates, because what you see is once you start to scale, parameters are changing, and actually the product is changing as well. So with that, so we're actually building two platforms. One we call Pulse. I will talk about that the majority today, which is actually our platform where we take cells from the donor, where we do reprogramming, where we do gene editing, and come with a product-specific master cell bank for the lifetime of the product. So, and we do that very early. So GMP constraints actually are coming in very early in the development process because we don't want to do that twice. We don't want to repeat that over again. So the rules of development are changing a little bit here. Then we have our Echo platform, which is completely bioreactor based, not for today, but I can talk about that during lunch. So such a manufacturing workflow looks like this. So we start with a patient or a donor. The donor supplies typically blood. We isolate the cell type of interest, simple magnetic separation. And then we have a very small scale reprogramming process where we actually reprogram a single cell into a single iPSC. It's not a, you work with maybe 1 million cells, but it's a very small scale process. And then you want to do your gene edits, and you want to do that in such a way that they're completely controlled. So again, you want to do it in one cell. So it's a very small scale process. So once you have identified the correctly engineered cell, you enter that into a cell banking process that might look something like the previous speaker with a cell stack. So you generate your master cell bank, and this can be hundreds of vials. Optional, obviously, for commercial production, you will obviously make a working cell bank like you do in every biologics process. And from there on, you do your drug substance and drug product manufacturing, ideally in stir tank bioreactors, because that's the most scalable way to do it. So when we start to think about an allogeneic iPSC cell therapy, and this is a highly simplified QTPP for an hematological disorder or an autoimmune disorder. So typically what we see is that a therapeutic developer is focused on something that is redosable, something that is off the shelf, transient and rapid. So rapid onset, but transient in the patient. That gives a deep B-cell depletion and no lymphodeletion. So this requirement set is now becoming actually quite a bit more sophisticated than sticking a CAR in a T cell. Typically you want to do more things, and that's what we have been seeing in donor-derived allogeneic. So yes, that therapeutic developer wants to put in a CAR, but might want to get rid of the TCR, which gives a safety signal. Might want to get rid of HLA class 1, might want to get rid of HLA class 2, and might even want to put in an NK inhibitory ligand to avoid that cell is immediately eaten up by the NK from the patient. So you now start to look at a very complex set of gene modifications. And the question is how to do that in a good process. So the question is how to build that multi-gene edited master cell bank, which is now the master cell bank for the lifetime of the product. Because it's actually quite a big risk to switch a cell line during development. I think you touched upon that. So ideally what you want to do is to do that editing once, create a research cell bank, and do your further process development from that research cell bank so that you can go back to that material and make the master cell bank from there. So an iPSC reprogramming workflow is actually really simple. So you do a source cell enrichment, you transfect or transduce those cells with the Yamanaka factors, you wait, and yes, I'm oversimplifying, you pick a colony and you do quality control. So one of the reasons that Mr. Yamanaka got his Nobel Prize so quickly was that this was broadly accepted within years. Within 2-3 years, I think every university in the Western world had an iPSC core facility that was doing this. So and so the field emerged really quickly. The problems are actually in later stages of the process. So then the iPSC editing workflow. So I think most people are now working with CRISPR technologies. They are precise, effective. So what you do, you design your guides, you bring those guides into the cell, you bring the CRISPR component into the cell, typically you get reasonable high efficiencies. You want to do a clonal isolation of the cell because you have an IC8 guideline, Q5D, that you want to respect. So you want to be sure about clonality. You select those clones and you do quality control on them. But now the question, if you want to do this 3 to 5 times, how are you going to do that? Because we wanted to make that QTPP with multiple gene edits. So the way we thought about this is we want something that is scalable. We want something that is robust. We want to be fast because we can't basically spend 1.5 years in a clean room to make a cell line. Efficient, cost-effective, well-documented, and regulatory friendly. And so basically, if you look at that and step back, so there are a number of quality parameters. Obviously, sterility, absence of mycoplasm, and the toxins and viruses are important. So that's kind of the basic, and I will stop talking about that from here on. What then is important is that you want to maintain a proper genome. So a cell replication mechanism is not perfect. So every time my cells divide, the replication mechanisms make a number of mistakes, typically 2 or 3 on a genome. So the moment that you culture cells for a very long time, you actually have risk. And FDA has just acknowledged that in their new guidance. They are draft guidance, I should say. They are talking about integration of all genome sequencing technologies for cells that have been expanded into a manufacturing workflow hundreds or thousands of times. And then you want to have a proper monoclonality of the cell bank. So we looked at that, and so on genome integrity, so we felt like the technology choice for reprogramming is super important, and for gene editing is super important. The timing culture I already touched upon. Want to restrain that as much as possible. We want to select the cells carrying the intended modifications in the best possible way. We want to do single cell sorting in a very good way, monoclonality assurance, and then obviously we come to multiple parallel clones that we want to handle. So how are we going to do that? So on genome integrity, reprogramming, I mentioned Yamanaka discovered iPSCs and it got quickly integrated. What we saw in those first years was it started with retrovirus, we went to lentivirus, we went to transposons, it went to adenoviruses, episomal DNA, minicircles. And so basically you saw from integrative to DNA-based technologies. Now we are DNA-free. For example, Sendai viruses, which will disappear from the cells with typically a number of passages or simple straight RNA. So those are the things that we're using on gene editing, first generation technologies, where obviously retrovirus, we don't want to do that. Then CRISPR came, typically plasmid based or virus based. We don't want to do that. So what we are right now doing is RNP, so ribonucleotide based. So we transfect it into a cell after 48 hours. Those proteins are gone from the cell. So what we have done in Cellistic, so we have integrated our proprietary CRISPR technology that we acquired a couple of months ago, searched the whole landscape and really looked for the best-in-class technologies for knockout and knock-in, because you can imagine the more efficient your genome editing, the less amount of cells that you have to screen. So what is nice is that we are seeing with knockouts right now, typically depending on the target, a 90% efficiency, and with knock-in, with optimization, we can get up to 50%. So that gives good numbers, and we can work with that. And then I think the most fundamental thing is how to manage that time in culture. So a single round of a gene editing will take you 3 months. If you go from 1 cell to 1 million cells, then you're already at 20 population doublings. A cell in our body typically does 50 to 70 population doublings. That's the Hayflick limit of when telomeres start to shorten. So we want to reduce that as much as possible. So we have to go multiplexing. And so when you start to do multiplexing, so you can do that with CRISPR technologies. So what you see with single knockouts here, you get these 90%. When you start to do duplex, you start to shrink a little bit. When you start to do triplex, you start to go lower. So meaning you have to start to screen more colonies. Simple math. So typically what we see if we do a triple edit, that we get to 30 to 60% insertions and 40 to 70% knockout. If you then start to do the math, you want to get at least 10 colonies out of it, you will probably have to handle 300 in parallel to have some safety margin. So then on monoclonality, so obviously we at some point we had limited dilutions. We had limited dilutions 2 times. It will take a lot of time, very manual, don't want to do that. So what we have implemented is this system from Cytina called the Upside. What is nice is that it works with 384 well plates. It deposits a single cell in a well. It images there, so to ensure that it's actually one cell. Also image from the bottom to ensure that there's one cell. So we have double clonality assurance, giving a lot of confidence that we are actually starting with a monoclonal cell. So on top of that, we actually image on day 0, on day 1, on day 2. So we get a lot of growth kinetics from that cell. So we document it very, very well from essentially the birth of the master cell bank. And so then to bring that together into a system, we again worked with Cytina. We call it a C station. That monoclonal liquid handler solution is inside. There's a liquid handler. It's actually shown on the right. You can hardly see that. There are 2 incubators attached. There's a robotic arm that integrates between it. It's a full BSL-2 enclosure in a clean room with scheduling software, with reporting software, and with imaging software. So we basically, I guess it's an islet of automation I just learned this morning. So that islet of information, so there's a system architecture behind it. So there are 5 pieces of equipment now integrated with a scheduling software, with software to create monoclonality reports. And so with that, we essentially actually give ourselves the chance to work with easily with 300 clones, because that's just one 384 well plate, or multiple if we want. What we also see is that therapeutic developers early in the development typically ask the question, okay, I have 5 different cars, which one do I want? So we can just build them all in parallel. So busy slides, in a way, what I'm showing here is essentially the application workflow. We have now transfected on the left our gene editing components into the cell. We do single cell deposition in 384 well plates. We monitor those cells. We pick the good colonies into 96 well plates. We passage them again in 96 well plates. We make a DNA plate. We do QC. And then once we know which one we want, we tell the device, okay, continue to culture those. And those will now be the basis for my cell bank, the master cell bank for the lifetime of the product. So with that, we have basically built our Pulse platform, where we allow our therapeutic customers to go from ID to edited iPSC, where we integrated proven technologies for reprogramming, gene editing, and cell banking. Because we are a new organization, we could basically build a complete purpose-built GMP environment where we implement automation from the beginning, where we have pre-established the complete workflows. We govern this under a completely dedicated quality system, where all the materials are screened by our quality team. Batch records are completely implemented, analytical testing completely implemented, and QA oversees everything. Because very early in the development, we actually want to build the cells in the proper way so that we actually have the right quality from the beginning. So I want to thank you for the attention, and happy to answer questions. Yeah, so so basically after the single cell deposition, we grow the cells again, essentially to that million that I mentioned before, actually a couple of million. We freeze a number of vials for quality control and also to generate R&D vials again. So with that, we essentially freeze maybe a million, 2 million cells, and that becomes the starting material to go into the master cell banking process. The current master cell banking process is built on CS2s. That can be a CS10 in the future, and so the moment that you are starting with 1 million cells in 2-3 weeks, you can make a master cell bank depending on the size of that master cell bank. So from 1 cell to 1 million is 3 weeks approximately. So an iPSC device has a population doubling time of around 16 to 20 hours.
Stefan, great talk, John Rowley, Rooster Bio. I thought, I've always loved, cell line development, and applying that to iPSCs is, I think, a really big opportunity. So congratulations for establishing a really nice platform. When you do your multi-gene edits, right? And it makes a ton of sense doing them all at the same time. But then, and then, I guess, how do you establish how do you choose the best clones? Obviously, I think you've got to start first by which ones even grow.
Mm-hmm.
But then how challenging is it to define the ones that are, with all the with the multiple edits that are the best?
Yeah, so indeed. So one parameter is how they grow. If iPSCs differentiate for whatever reason, you can see that quite easily. So an experienced operator sees that because iPSCs are very compact, and the moment that they start to differentiate, they start to get larger and they grow. So we don't want that. So that's the first pass quality criterion. The real screening, what is the right clone, is based on PCR-based methods and sequencing-based methods. So the knockouts we determine by sequencing, the insertions we also determine by sequencing. We also look, I didn't mention that, we also look at off-target effects, because based on bioinformatic prediction where those guides can also cut, and so we do that relatively quick, so that we basically have all the right information to only progress the colonies that have the right modifications according to our specifications.
So I don't do it, I'm having a hard time fully visualizing it, but I guess how for each single clone that you do move forward based on growth, it doesn't grow and as long as the state is differentiated, how do you have to get it into T-flaps before you can do all of the choosing which one is best, or
No, we
can that be done at a pretty small scale?
96 well plates, we do that screening. Yeah.
Awesome. All right, cool. Yeah, nice job.
Any other questions for Stefan? All right, hearing none, we have a break now until 10:30. I encourage everyone to continue the conversation during break, and we'll reconvene in this room at 10:30. Thank you.
Catalent is a service provider. We have 90 years experience on different modalities, product development and manufacturing. Currently, over 1,400 drug development programs ongoing in different modalities. We've in this space experience with over 100 cell and gene therapy programs. In cell therapy specifically, we have experience for supporting 20 programs, and mainly autologous and allogeneic cell product development work and manufacturing. Since the first launch of the product CAR-T, Kymriah, chimeric antigen receptor T cell therapy has been in this space as a new family member, helping patients and then treating patients with mainly acute lymphoma and then over 6 commercial products have been right now always the pharmaceutical company saving a lot of people's life. So in particular, that autologous CAR T cells has the need for the growing volume, as we're saying that the growing numbers of the cell therapy programs in going into the late stage clinical trial for commercial. In addition to that, there are the autologous and allogeneic, programs in development work from preclinical stage all the way to the late stage and commercial. There are the expansion of the indications now supporting patients with solid tumor with fantastic clinical outcome coming from the CAR T cell technology as well. And outside of the oncology, now indications are expanding to autoimmune diseases. As you know, that is a big patient population. However, the challenge is slightly different for CMC. Overseeing those programs going through early stage and now late stage commercialization coming around the corner. And then you can imagine that's going to be, even for some indications, going to be over 8 million patients per year. So that's sort of, the future forward thinking, how we can, using now best-in-class technology and how we can improve on the manufacturing efficiency to support that type of, volume coming to not necessarily, staying with the current status will help us to address all those challenges. For now, today, autologous is still the front runner and CAR T most prominent approach. iPSC pose really great promises for both autologous and allogeneic regenerative medicine in this space. And then we also have some internal work to do for the iPSC platform. Leveraging our existing experience supporting over 20 programs. So we launched the uptempo CAR T platform to speed the path to patients. So as you know that we have experience now in the field, 6 commercial products, really know how to make and manufacture the product. But in order to help the developers and the companies, not necessarily to redo the work, but leveraging the existing experience. So we built the ecosystem using best-in-class GMP compliant the cell therapy manufacturing equipment automated based on each unit operation. And then we have also onboarded automated analytics on this platform ready to use with streamlined templated manufacturing draft batch record protocols and SOPs. So experienced scientists are supporting the process transfer and onboarding those process and analytical elements to the manufacturing facility, clean room and facility. Then we can get really reduce the startup time and reduce the time and cost for the start of the clinical trial. We are offering that and to really partner with early stage companies and to accelerate, the product development path to patients. I mean, at the end of the day, patients are our customers, and also really, this will be helpful at least for now to support some of the amazing programs in allogeneic and autologous space. So the process starts from the clinical side for autologous CAR T cell products. we collected the freeze sample and ship over to the manufacturing. The cells can be started to processing it right away if cryopreservation can. We also have the protocol to support the cryopreserve the starting material, isolating of the PBMC and followed by the CD4 the selection of the T cells. And then the cells will be activated by Transact and transfused in bag with a lentiviral vector, with or without the lenti-boost. And then we have already done the comparison work for different bioreactors for cell expansion between day 3 to 7. And after that will be cell wash using CPAX, Lovo as a backup, and also can do automated fill and finish, including Q, Finia, and then the product packaging, and then shipper to send to the clinical site for treatment. Wanted to share with the development work, proof of concept, and then when we develop this, the team has done the fantastic work, kind of thawing the bag, and then and then using the plasma serum to transfer the cells into Climax Prodigy and for the automated cell selection. We've done the comparison between Plus and Prodigy, and Plus is sort of still a little bit manual. That's the thought process, but still, the two options are open, but the idea is to close the process and automate on each unit operation. So we selected the Prodigy, not necessarily the still we're improving, but this is a sort of the 1.0 process. And then after that, we do the G-Rex and the culture bag for the activation. As you can see, the sampling pan inflated using the automated flow cytometry Cellistic for in processing sample testing and pre-selection and post-selection for the enrichment of CD3. Overall, the recovery is greater than 70% for the starting material selection. So next module is cell wash transduction. We wash the cells and then using CPAX to wash the cells, and after that it will be transduction using lentiviral vector and lenti-boost if that's being used for the initial process. And then we do the transduction in the incubator. And then we actually, for the process characterization, done the parameter ranging studies, and then to look for the range of this cell wash and potential loss or recovery of the cells. Transductions, I want to highlight this, and it's interesting that for our CAR T cell products, you still need to use the vector at the moment. So the vector design part, it's interesting. We have a gene therapy group helping us to do the in-house vector design and then development work. So what we are collaborating and then gets also, Catalent wider network to do for us is to kind of design the vector where we can accelerate the timeline for using the vector for proof of concept and also for the functionality testing before it goes to the clinical grade vector production. So that's the team was doing. I'm sharing the data showing that we have a CAR and then we have a tag where here in this case is a GFP. So it's on the same construct, and then we transfuse the primary T cells isolated from healthy donor, and then we did MOI study to look, at mainly it's a sort of the volume you wanted to identify appropriate for the clinical manufacturing. So as you can see that as you're increasing the concentration of the vector in the cell culture, And then there's going to be a plateau, right? And then over there, it's, over the plateau, and then everything you add more, it's all about the cell and the vector ratio. All you wanted to identify is that the plateau, and then the MOI 5 or 1, and then that's the volume you wanted to use for the clinical manufacturing. Also, you can see in this case, the GFP and the CAR expression are 100% correlation. That makes sense because it's on the same construct, and you want to insert that vector into your genome, and then on single cell level, it should be 100% correlation. So that's sort of the example I'm presenting here, sharing the work we're doing. This one is an important arm to integrate into our CAR-T platform. We've done the bioreactor comparison for cell expansion. Existing technologies, quantum, G-Rex, and Ruri. So they can be plugging, swapping it out. But we also compared those technologies for the cell expansion and cell health. Sharing the example over here, comparing quantum versus G-Rex, and expansion is better on the quantum instrument over a 7-day period of time. Viability all great, over 85 or 90. And G-Rex, we've accomplished 40-fold increase over 7 days. Look deeper into this data on the right-hand side for the quantum lactate generation, glucose consumption. You can see, over 7 days, and then the increase of glucose consumption and lactate production really aligned with the cell expansion, suggesting the good cell quality over the expansion period of time. Also recovery, I want to point it out, our data showing only 77% of recovery, probably is still above the acceptance criteria. Not 100% recovery, not going to achieve that, but 77% is still, I think we have some room to improve for the recovery for the quantum. You don't lose much cell from G-Rex and Zuri, so therefore, our recovery is always over 90% from the 2 other devices. So combining the expansion fold increase and also the recovery, at the end, calculating the total number of the cells for DP, we've seen a very small difference actually now between quantum and then G-Rex and Zuri. So therefore, there's some room for improvement, absolutely for Harvest, but at the moment, really, I think in particular, quantum and G-Rex both are viable options for clinical manufacturing. Harvest and fill finish, trying to automate this piece, for autologous, it's only 2 bags, sometimes it doesn't, but if you automate and close the system, that's a huge difference for production. For the clean room setup, the draft batch record, and you can automate that piece. Therefore, the team did the comparison on the technologies between Q and Finia. So the maximum bags you can fill for Q is 8 bags, and Finia 4 bags, right? Like if you do 4 rounds, that will give you 16 bags. So for the Finia, still acceptable for the cryo, for the cell sitting in the cryo media. Comparing to the Q, the Q also can do the cell wash. If the volume is not big for autologous, the volume is not big for allogeneic, that could be a challenge for that volume to wash the cells. So therefore, for autologous, again, 2 bags, so both are viable options, but what will make sense if we want to extend our work to accommodate allogeneic programs where we have. So therefore, we're still evaluating. I think, at the moment, the team's thinking the Q can be and then a recommended approach. But we're still kind of evaluating potentially next generation technologies to accommodate both autologous and allogeneic backfill and vial fill. So that's, we also again mentioned the analytical QC packages, all those methods available, not necessarily all qualified because we do need the product specific qualification on those methods. But really, like I would say, I've been in this cell therapy field for a while since I was doing the CAR T Kymriah work. So all those methods, I think, for this audience could not be like a surprise for you. It's really standard. It's a matter about the time and cost and how when you can get started for the clinical trial, material production and the release. Summary of the CAR T cell platform work. We've launched the modular design of the up-tempo CAR T manufacturing workflow, which is a modular design. It's flexible enough for the customers to select any unit operation and to put in your process into this entire workflow. And then it's a GMP compliant it's closed system. And on the side, we have a product development team, both in Princeton and New Jersey, and also in Europe, and help on the QBD design of experiments, and then to optimize the parameters for different cell therapy programs and specific needs. And also we're supported, of course, by the Catalent wider integrated experts and facilities. In particular, our quality system is kind of overarching across multiple facilities. That will make sense. And also, harmonizing the regulatory needs, from different regions and things like that, that could be helpful. But it's outside of product development. I just want to mention in those potential benefits. For the iPSC work, it's our thought process is it's really complex product and then process. So we work on, again, different units. For the starting material, the team has done the work, and then to calculate how much time we need, it's 6 to 8 months to customize a donor cells and reprogram those to the customized GMP compliant iPSC banks. Or we have already existing GMP banks from the US donor and from a European donor. Those are all available for cell banks. So we have research bank and we have GMP compliant, so you can easily get a research bank and then to do it in your lab and then check on the cell quality and then the differentiation potential to your target tissue. That was a thought process, providing a very flexible platform for the customers to use. On the right-hand side, we have genome engineering. So I'm a little bit running out of the time, so I'm going to be sharing these slides, after the talk as well. But the genome editing to create the hypoimmune cell banks, and the team will present at ISCT. And we can do also differentiation to different target tissue. In particular, I want to mention that we have the CAR-NK proof of concept data from the product development team. And we're using both donor material generated NK or use iPSC derived NK. And then, again, the persistence of NK for treating those patients. So linking to our CAR lentiviral capability, we could do the CAR-NK development work. Of course, the proof of concept for how this would make sense, in terms of the cost and efficiency. So the team now working out the GMP compliant protocol and using the high quality intermediate, which is iPSC derived precursor cell. Those are cryopreserved, so can be easily to give rise to the NK and for the scaling up. We've shown the proof of concept, the mature NK phenotype Of course, you do see the purity marker expression of phenotype. You don't know whether that could kill the cancer. So what we did is a cell-based IAC co-culture the NK with the target cells and then measure the NK. But the interesting finding is between the two media we are testing, one medium could potentially expand the cells, and then the other one is kind of helping the cells to kill, maintain that killing efficacy. So we're doing actually now, including the media optimization, you can see this is sort of the data showing that if we're swapping back the media, and then that potentially can change the functionality of the cell back from the killing cells to the expansion cells. So I guess, in a very simple way to explain this is the cell would require energy either to divide or to kill. So, one medium formulation promote the cell to divide, and then you get a very good expansion. And the other formulation gives you a better killing, and then the cells are focusing on the killing using their energy and not necessarily dividing at the same time. So just a sort of my way to explain that, but we are doing also optimization, summarize of the iPSC platform work. The work has still some room for improvement, but we have GMP compliant iPSC lines to offer for allogeneic cell therapies. We've differentiated into different target tissue, including hepatocyte, cardiomyocyte, MSC, NK, pro T cells. Those demonstrating the cell line can have a great potential differentiation to that target tissues. And we're optimizing the processes, including the media for the cell expansion. I'll pause it here if you have any questions. Thank you.
Thank you, Junxia. Unfortunately, we're out of time and we have to move on to the next speaker. I would like to welcome Eytan Abraham from Resilience. Thank you. Also, a reminder to all the speakers, you have a timer on the podium. Please pay attention to the timer. Thank you.
Thank you. Timer is very visible right here, so I'll take that into account. Great being here. Thanks for the invitation. So I'm going to cover a couple things, if the slides come up. There you go. Thank you. I know how to do this. Yes. Okay, so want to be a little bit provocative here, right? So right now, as an industry, we're around 100+K for cost of goods for a lot of the autologous cell therapies. That's not sustainable. We can't reach many of the patients who need these therapies. So a lot of discussion has been over the past few years, how do we get the cost down? A big piece of that is automation of the manufacturing process, but there are lots of other pieces we can leverage. So actually, I'm not going to be talking at all about automation of the manufacturing process. I'm going to talking about other elements that we can use to reduce cost. So first, couple words about Resilience, because we're a fairly new company. So 4 years ago, we want to do a lot. So we are doing a lot. I'll show you our network in a moment, but it's end-to-end capabilities in terms of biomanufacturing, excellence in biomanufacturing. Also a lot of investment into innovative technologies that will facilitate types of biomanufacturing we need for these advanced therapies. And also differentiated business models. I mean, we acknowledge that, especially in the current environment, many companies have cash flow challenges, let's put it that way. And how can we, as a service provider, work to defray some of those upfront costs of getting therapies, especially to first in human. So we work on a bunch of different modalities, spanning biologics, vaccines, drug product on the established modalities side, and then nucleic acid, cell therapies, and gene therapies on the emerging modalities side. In terms of our network, we have a pretty extensive network, especially in North America. And that's where the name Resilience came from. The origin of the name in the company is to see how we can bolster initially local manufacturing in North America to make sure that there's assurance of supply chain and robustness. So as you can see here, multiple sites working on multiple modalities. Our main site for cell therapy is outside of Philadelphia. And in that site, we have multiple programs running, including a pivotal program. That site is on track to becoming a commercial site. And our main viral vector site for GMP is North Carolina. It's a legacy Bluebird site. And that site actually just got recently accredited for commercial manufacturing for viral vector by the FDA. So a lot of activities. Our main site headquarters and site for main R&D activities is in La Jolla, San Diego. So this is one aspect of what we work on, is how do we combine these different modalities to get therapeutic developers what they need. So starting from plasmid, how do we source efficiently and in a good fast turnaround and good pricing through nucleic acids. A lot of cell therapies now need the incorporation of certain nucleic acid elements, mRNA and others, guide RNAs, obviously viral vectors. So how do we combine all of these things and try to source them as efficiently as possible, either in-house or externally, to get the therapeutic developers what they need quickly and easily? I know it's very difficult to manage a whole bunch of companies that supply what you need. So one other element that we feel is important is how do we build what's needed to supply all of this, but also additional elements, not only the materials, but also, as I mentioned earlier, the funding, the clinical access, the regulatory support, and the biomanufacturing, right? We do a lot, but we can't do everything. And the question is, it takes a village. The question is, how do we make it easier and faster, especially for early phase companies that might not have the time or the resources or the manpower to work through all of this? So how do we build partnerships to enable all of this or most of it? So I'm going to get to the crux of this talk, which is basically how do we move down and improve significantly the cost of goods. So we do this day in and day out, so we know what this looks like. A 7 to 9 day manufacturing process for a CAR T. COGs are round about $100,000, north of $100,000. So what does that look like? So we have here several things. So we have FTE costs, right? That's the main cost, and it's basically 57% of the cost. FTEs, raw materials, about 25% of the cost. Now this is with a mostly closed, somewhat automated process using Prodigy Infinia, but obviously we can use more efficient platforms as well. Then there are a few things that further drive up costs. So one thing is variable harvest days, where a lot of these therapies, the harvest day could be 7, 8, or 9 days, for instance. If you have a variable harvest day, then that drives up costs significantly because you have to staff to be able to harvest at different days. And then accelerated turnaround time for QC. We talk a lot about manufacturing, but what happens when we have to do a 2-week, extensive, complicated QC release with multiple pieces of equipment? I'll touch on that later, but trying to accelerate that by throwing people at it, throwing equipment at it, is an additional cost as well. And turnaround time is critical for the patients, obviously. So I'll be talking quickly about 3 elements here. So one is viral vector, right? Is that viral vector is a significant cost of the manufacturing. How do we bring that cost down? But also, how do we reduce the turnaround time to get from gene of interest sequence all the way to a GMP batch, especially for first in human? The second is going to be how do we shorten the manufacturing process? So again, one of the easiest ways, fastest ways to significantly impact COGS is to reduce the manufacturing time, right? Because you can basically cut your cost in half, roughly, especially from a FTE standpoint, if you reduce the time frame. So we've developed a 3-day manufacturing process. And then last but not least, our push to basically replace almost all of current QC methodologies for release using NGS-based platform. And then ultimately, how do combining all of these 3 elements impact cost of goods? So starting with the viral vector, so you can see here several examples of options we have, right? It's all about optionality in terms of supplying viral vector for different modalities, whether it's cell therapy with lenti, whether it's in vivo gene therapy with AAV, but all the way from a standard offering, which encompasses full development, analytical and process development, up to 2 times 1,000 liter manufacturing, et cetera, through what we call our essential offering, which is truncated and focuses more on 200 liter, but also reduced activities and reduced timeline. And then to the last one here, which I'll talk about, which is what we call SNAP LVV, which is basically fit for purpose for phase 1, under 12 months, with a low fixed price that includes all the raw materials, including plasmids, and a 25 to 50 liter bioreactor scale, which should get you, obviously, depending on the yield, depending on the MOI of the therapy, of the manufacturing process, should get you sufficient material for your phase 1, right? So how do we use that specifically to make it easier, faster, and cheaper for early phase companies to get into first in human and conduct their phase 1 trial? So as I mentioned, this is the goal of this is really to be a quick to clinic, serum-free, suspension-based lenti process that gets the therapeutic companies what they need. So this speaks a little bit about very high level about the process. Basically, the advantages are it's this one batch, which is usually enough for a phase 1, a continuity of cell line when you progress beyond your phase 1, continuity of cell line plasmid, and analytics, so that you can scale and make this larger volume for later phase and commercial. And then also quick release. So again, from gene of interest to GMP batch release in less than 12 months, bundling the raw materials, et cetera, within the cost. Quality review that is fit for purpose for phase 1, and a path to commercialization. So basically, how do we build something holistic that is fit for purpose? So just in terms of the performance, again, just a snippet here. I'm not going to go into too much data, but you can see here good performance, good responsiveness to increase of MOI, et cetera. So a good, robust lenti process that yields what's needed for first in human. The second topic I'll discuss is the manufacturing process, right? Or more specifically, the shortened manufacturing process. So this is sort of our current standard manufacturing process and the equipment we use. We're assessing other types of equipment, such as the Ori solution, and we're definitely open to other solutions, but this is what we're currently using. So obviously, utilization of Prodigy, if and when we need higher cell numbers for some types of immune therapies, also using the Zuri as a solution, and then using the Finia for closed fill finish. And by the way, the main approach we're taking to manufacturing in Philadelphia is a ballroom setup. So ballroom setups, I think, is the future. That's where things are going to go when they get to commercial stage, not small B-grade clean rooms. And that's where we need to be in as early as possible, both in terms of the manufacturing platform solution, as well as in terms of the build of the facility to make it an easy transition to late phase and commercial. So as you can see here, this is sort of the process. And we can do fresh or frozen leukopaks, et cetera. Fully digital platform, digital batch records at the site, MES, et cetera. So quite a robust system. But the question is now, how do we take this platform, take this system, and we reduce the timeline? So usually what we see is when we look at CAR T manufacturing is that as we get more cells and the dose size increases, we're getting a reduction of activity of the cells, right? We get an impact there. So how do we cut the manufacturing process down, not only to reduce COGs, but potentially to be able to use a lower dose for the clinic because the cells have a better functionality. So that's essentially what we did. And again, I'm not going to get into too much detail here. Part of it is obviously proprietary and confidential, but basically we take a bunch of these steps, we significantly reduce them. We move from a 8 to 14 or 7 to 14 day process to a 3 to 4 day process. Obviously, there are many challenges associated with this, but the data, which again, I'm not going to get into too much, shows that these cells are robust. It's repeatable. The process is repeatable. We get good results both in development lab as well as in the GMP space. The cells are pure, they're highly functioning, and they retain a good central memory phenotype. Just in terms of expansion of the cells, we see very good repeatability and good expansion. And regardless of the fold increase that we're getting, the range of fold increase we're getting, we're always getting to the target release batch size or cell number that we're looking for. So the last element that I'm going to discuss briefly here is the QC platform. So as I mentioned, right now QC is complex, expensive, very labor intensive, very space intensive, and just with a long turnaround time. So how do we address that? So there's some elements that are being addressed, like using digital PCR for some of the release assays, et cetera. And this is also a challenge from a regulatory standpoint, obviously, because the regulatory bodies need to agree and approve to change some of these standard release methodologies to another approach. So our approach is, how do we take basically all of these? And you can see here an example of the range of different types of equipment needed to release a CAR T therapy. And we basically replace this all with essentially one system. So that one system we're working on, again, is automated for the sample prep. It requires a much lower volume because it's NGS based. And when I say NGS based, and I'll talk about this in a minute, I don't mean just looking at DNA or just looking at RNA, but also looking at protein. And the way we look at protein is by conjugating antibodies to DNA sequences so that we can use the NGS method to measure also protein, not just RNA and DNA. So what we're looking at here is basically moving from whatever it is now, 2-3 week release to basically a 2-day release, 48-hour release for all of the assays, obviously generating a holistic report that shows that everything is released properly. And then this is, as I mentioned, this is for things like mycoplasma sterility, but it's also for immunophenotyping, vector copy number, cell viability, et cetera. And by doing this, we estimate that we will be reducing QC needs by about 95% for FTE time, 60 to 90% of release timeline, 90% laboratory space, and 80% CapEx. So very significant reductions in all of these arenas. So here's just again an example of a comparison between traditional flow cytometry and what the NGS-based method can give us. So you see that it looks very similar, but the added benefit is we get a lot more granularity in terms of understanding the cell populations within the culture, which you can see on the right here. Another element here is, especially with a short process, how do you track and verify your CAR positive cells, your CAR expression, right? Because it takes time for that to be expressed on the cell surface. So again, using this NGS-based method, we can very much earlier detect what the correct number of cells that are CAR positive. So that also allows us to release accurately a product, even though the manufacturing duration is significantly shorter. So to finish, in conclusion, right? So taking all of these three elements, which again do not encompass really some of the novel ideas around automation and manufacturing, but taking these three elements, we get to a very significant cost reduction. So we get from north of $100,000 per batch to around $40,000 per batch, which is huge. FTE costs move from 57% to 43%, but more importantly, they're reduced from $68,000 to what is this, $17,000. And raw materials also are reduced significantly, both because of the efficiency of the lenti component, but also just because a shorter process requires less materials. So material costs go down as well. So the drivers for FTE reduction is obviously the shorter process duration, the use of the CONA system, the NGS-based QC system for QC release, elimination of a variable harvest day. And obviously, we can also get more batches from a given clean room space because the manufacturing process is shorter. Therefore, the overhead that we're assigning to each batch is reduced as well. Then in terms of raw materials reduction, I mentioned the use of the lenti approach. and the shorter process. Now, there are additional gains to be had here, right? As I mentioned, more automation of manufacturing and further reduction of raw material cost of goods. So 40 is not the lower limit. We can go below that, and we need to go below that. But in this talk, I'm addressing other elements here. So thank you, and happy to take a couple of questions.
Okay, we have time for one question.
Great, so it's crystal clear how we get to 40K. That's great.
Eytan, I'm not sure I saw logistics costs in there. And if you're even in a regionalized model where you're shipping, starting material and final product back for.
Yeah, I'm talking about dock to dock. So it starts from when the leukopak arrives at the site, and it finishes when it's about to leave. But definitely shipping costs are an additional cost, and I think those also over time, especially with volume, we're going to get to better numbers. And obviously decentralized. If we whatever that is, whatever definition is, shipping costs are going. Yeah, we'll sort out exactly. We'll sort out later. Yeah.
Great, thank you,
Thank
Eytan.
you.
Yeah, I'd like to welcome our next speaker, Jason Foster from Ori Biotech.
Hello, everybody. I'm Jason Foster, the CEO of Ori Biotech. Ori is an innovator in the cell and gene therapy manufacturing technology space. I think Eytan's presentation and mine will show that there is a fair amount of consensus around what's driving current costs, but maybe a differential understanding of what those costs actually are. And I think I've seen lots of different estimates. That's on the low end of the ones that I've seen, at least. So let's talk about what goes into really understanding. If we can at least pushing all the buttons here. If we can understand what's really driving costs today, then we can really address them in a meaningful way. I always like to start out by focusing on what we're doing right, because we spend a lot of these sessions talking about all the things that aren't going as well as we want. We had the pleasure of meeting Opie Jones and his mother, Lucy. Some of you who were at ARM meeting on the med might have met Lucy as well. Opie was one of 9 children in Europe to get access to what is now Kymriah in 2021 when he was diagnosed with ALL and is now approaching 3 years cancer-free, which is an exciting milestone for him and for the industry. Unfortunately, Opie is one of the lucky ones. We are only able to treat a very small proportion of the patients who could potentially benefit with today's therapies. And that's not getting easier as we launch more products. The complexities of treating patients become even more significant. I've also had the pleasure of meeting Amy Kappen, whose daughter unfortunately passed away, Sophia, after receiving CAR T therapy very, very late in her treatment and relapsing off of the standard of care. Amy has started a foundation called the Best Day Ever Foundation to support patients and their families that are going through cancer treatment. We have to realize that for every success story, there's many, many stories, unfortunately, like Amy and Sophia's. I asked the team at Ori one day, how many patients are getting treated? And everyone looked at me sort of blankly and said, well, we're not sure. And I said, well, can we go find out? And they said, well, we've worked on this for weeks and weeks and can't really find the numbers. So we went out and we tried to create these numbers off of publicly reported information. So at least we can track progress. We can track trends. It's probably exactly wrong, but directionally correct, the numbers you see here. These are based on the revenue numbers divided by the price per dose of each of these therapies, which is public information. It should reflect roughly where we are as an industry. Last year, we treated roughly 9,000 patients, up about 37% from the year prior, which is great for those 9,000 patients and their families. That does leave quite a lot of people, unfortunately, without access, particularly outside the United States. And if you look at the Q1 numbers, you're seeing flattening in the trajectory of Kymriah, which has been consistent over the last several years, but also worrying drops in the availability of Abecma and some of the other products that were growing quite well. So we're continuing as an industry to struggle with manufacturing, and it's something we've been talking about for the last 5 years. And I don't feel as though we've really fixed the problem yet. And why is that? Why is it so hard? These are personalized medicines, as we know. They're very hard to manufacture, very manual. They're very expensive to make, and we're going to discuss exactly how expensive we think they are. But also those high cost of goods, the variability of these processes, high failure rates, low reproducibility, lead to, let's say, unattractive economic profiles for many of these products. And I would argue that as an industry, we have to be very focused not only on safety and efficacy during our development, but also on viability, commercial viability. Can we make these products? Can we launch them at a reasonable cost of goods and charge a reasonable price to get market access? The answer today is universally, that's pretty difficult to do. And we're trying to see how we can do that in a better way. Slide, getting excited there. Okay, perfect. So if we can at least today succeed in establishing a baseline to say we all understand the drivers of cost of goods when we're developing products, that's a reasonable place to start. I would venture to say we have a differential understanding of that today, and everyone sort of kind of views this issue a little bit differently. So what are the current drivers of costs for, let's take a CAR T therapy today, because that's the lion's share of the approved products. I think Eytan was 100% right. A lot of that is labor. A lot of that is consumables. The vast majority, 60, 70% is labor inputs and reagents, viral vector, consumable sets, single-use materials that drive much of the costs currently. We also have, for the majority, paper-based processes. So we are running around with paper lab notebooks, paper batch records, or paper on glass. None of that is particularly helpful as we think about efficiency and cost. To the point that was raised also, we also are thinking about replicating. When we try and scale, we replicate an inefficient process, and we continue to do that without trying to fix the inefficiencies. So our facility layouts and our facility design processes need to be optimized for throughput and for cost of goods, which we haven't done yet, and which is really causing complexity. That causes those facilities to be massive, 150 to 300,000 square feet to manufacture a couple thousand doses a year. That's expensive to build, 150 to 300 million to, it's about $1,000 a square foot to put that kind of facility together. Plus you've got the ongoing CapEx of that, 30 to 50 million per year just to run the facility, just to keep the lights on. And that includes very highly graded clean rooms. We want to be in grade C and grade D, but today we're in grade B with isolators for the most part. And that also includes offline manual analytics, expensive reagents. These are the drivers. These are the things that are causing us to have very expensive cost of goods and a resulting very high price and limited access. So what's the number? Depending on who you believe, it's somewhere between 100,000 and 270,000. So the top two boxes here, you see there's a recent report from ZS Associates, peg the number somewhere between 215 to 290. Lyric pegged it at 270. We have a number of our own, and this shows you the kind of range. Now, there's a difference today between cost of manufacturing and cost of goods. What Eytan was just referring to, I would call cost of manufacturing. What happens from dock to dock inside the manufacturing process. But there's a lot of other costs associated with apheresis, chain of custody, chain of identity, cryopreservation, logistics. And I think some of the estimates that are on the higher end are really focused on including everything from end to end. So when we talk about this today, I'm going to look at roughly exemplar processes. So these are Zuri, G-Rex-based processes that are using standard equipment like the Prodigy to do selection, the Lovo to do wash concentration. You might substitute the Cephia or the Cepax or the Quantum in some of these places, but effectively the Rotea. These are the processes. These are the pieces of equipment that are going to be used today. This is what I would call a representative process. And if you look at the top line, this is pretty close to the process that UPenn tech transferred to Novartis back in whatever that was, 2015, 2016, that's still being used in a lot of places today. If you look at that workflow, the bottleneck isn't wash concentration, thawing, selection, or downstream fill finish. The bottleneck is transduction, activation, expansion. That is the problem. And if you look at those estimates from Lyric and from ZS, they assign about 40 or 50% of the cost to those two steps, transduction and expansion. It's really that bottleneck of 5 to 7 days that's preventing our ability to increase the throughput of these processes. So just replicating this process over and over again doesn't fix the problem. We have the same bottleneck, we have the same costs, and the same low throughput and challenges. we need to fix the process before we start scaling it. And so focusing on the current bottleneck is where Ori is really focused its attention. So what I'm showing you here is our estimate of a fully optimized Gen 1 process. So a process being used today that's fully optimized to produce 10,000 doses a year would cost somewhere in the $160K per dose. And that there aren't any today that are fully optimized. This is a process that if it was fully utilized, what it would look like. And that's roughly about 50% consumable costs, so viral vector, activation reagents, others. It's about 40% labor. And if you look to the middle there, much of that labor is skilled labor. So highly skilled labor. To produce 10,000 doses, we'd need 5,000 people, of which about 3,300 of them are highly skilled operators. We know that that kind of, the economics of that just don't work. It's a very, very difficult value proposition. But that is really where the industry is today. Plus, we're using, as we said, a lot of very kind of high grade, grade A, grade B spaces to do this work in. So what's the alternative? What's the benefit of automation in this kind of current workflow? You don't have to scrap everything and start over. You've characterized a lot of the steps of this process. You understand them. They've got clinical track records. But we need to fix the bottleneck. How do we get the best of both worlds? How do we able to create a best of breed approach versus a jack of all trades, master of none approach, which we seem to have with some of the first generation technologies. So addressing that bottleneck is where Ori plays. So we're launching in 2 weeks' time at ISCT. Hope to see all of you there. And please, if you want like a demo of the system, shoot me an email. We'd love to show you what we've been doing for the last 5 years. But ultimately, the Ori system has been built bespoke from the ground up to fix the manufacturing, the flexibility and scalability challenges that are facing us in cell therapy. That means there's an instrument, there's a proprietary set of consumables. Those instruments can be multiplexed. They can be stacked effectively. So we can do 1,000 doses per year and 1,000 square feet of clean room space without changing anything about your process besides what's in that checkered box right there. Fixing that bottleneck massively increases the throughput, decreases cost of goods, and increases reliability. And we can do that using our automation technology. So how does it play out in the numbers? The numbers effectively, we 5x the throughput of that same footprint. So when we think about, we've got a total cost of goods model, which is essentially what this outputs. We take your process, we take the labor, we take the size of your facility, we take the reagents you use, we take the equipment you use, we put it in the model, and we can give you a very accurate picture of what your cost of goods are going to be. So that you know the process that you're designing about where you're going to land from a cost of goods perspective, but also importantly from a throughput perspective. You're going to have a throughput target that you're trying to achieve with this process, supplementing, sort of replacing the existing activation, transduction, and expansion technologies with the Ori technology would 5x your throughput in the same amount of space. It would also dramatically cut your cost of goods from today was probably around $160K to under $100K. And that most of that saving comes from labor. You can see a large reduction, 60% reduction in labor, but also a significant reduction in consumable costs, fixed costs, and also some of the paperwork and traceability aspects. So that's a great first step. And this is at a fully optimized 10,000 doses a year. Again, even Kite, who's best in class, is producing only 4,000 doses a year. So the idea is we're getting economies of scale here at these numbers. We're not getting them at 1, 2, 3, 10 doses a year, where we also need to be able to do cost-effective manufacturing. So the impact on labor, dropping that 5,000 number to about 2,000, dropping the amount of space by about 75% from a clean room perspective. You don't need nearly as many clean rooms. You don't need nearly as much space. You don't need nearly as many people if you're able to implement the Ori system into your process. And that means overall your facility shrinks massively from a couple hundred, there's 1,000 square feet to maybe 150, with the vast majority of that being warehousing, office space, QA, QC space. It's a space for all the other things that you're doing, not necessarily the manufacturing. And this is in a 1,000 square foot footprint that continuously gets replicated. We've limited to ourselves in that way so that we can think through the implications, because if you have ultimate permutations in every size facility and every type of process, it's hard to put numbers around. So this is in a 1,000 square foot, essentially replicating an optimized process in that footprint. However, working with partners, we have designed an optimized, let's call it a ballroom footprint, which Eytan just referenced, we think we could get 7,000 doses out of 2,500 square feet, which is a massive improvement upon what I just showed you. But it's by optimizing the use of each piece of space for the right grade clean room, the right number of people to get the highest throughput you can. Now, this is just an example. We would do this work with you specifically with your process to understand what an optimal layout and throughput look like. What happens next? Once we fix that bottleneck and we get to 1,000 doses and 1,000 square feet, what else happens? How do we fix the rest of the problems? Because ultimately bottlenecks will pop up downstream. QA and QC, batch release, sterility testing is going to be a massive bottleneck if you're operating at that scale, and upstream manual operations like media prep. So Ori has the ability to, in our R&D pipeline, to innovate at other unit operations as well, to combine those pieces to solve those problems. We thought it was very important for us to solve the major problem first, because if you try and do everything at once, you will always get a limitation. You'll be good at some things, but not at others, and you won't really solve the problem. So we're trying to retain flexibility for our partners and really fix the major drivers of cost. When you fix those other bottlenecks, you again get a leapfrog forward in efficiency. So you go from maybe 1,000 doses a year to 2,000 doses a year in the same footprint. You go from what was 100,000 or so of cost of goods to about 75K, again, dramatically reducing labor almost to zero, not quite, but also impacting all those other pieces. You'll notice that actually consumables becomes a major piece of the puzzle here. Once you get down to that level, you've got those consumable costs are pretty much dominating. You've gotten the labor cost out, you've gotten a lot of the logistics cost out, you've gotten a lot of the other costs out, fixed costs and others, but we haven't been able to adjust reagents and consumables and also some of the logistical pieces. So how do we go from 75K to something lower? And really that relies on, as Eytan was presenting, more efficient vector manufacturing. If we can get vector costs from 40K to 10K or less, that has a massive impact, or potentially using transfection instead of transduction could drop that to close to zero. And opportunities like decentralized manufacturing. Ori's is a cloud-native IoT platform. You can see into it in real time from anywhere in the world. Potentially, you could do quality checks centrally if you're doing manufacturing in multiple places, which is often one of the knocks against decentralized manufacturing. How do we release products? The ability to do some of these things gets us to that 40K, which we think is a reasonable target to have in the next 5 years or so. That was an express tour. So what's the low-hanging fruit? The low-hanging fruit, in my view, is automation. Take the people out so that we can take 50% of the costs out of the process. With the Ori system, we take 50% of the labor out roughly and cut the cost of goods by 30 to 50% of your current process. Automation is the first step. Automation will help bring more capital into the space, continue investment, continued competition. We know how pharmaceuticals work. Multiple products launch into the same indication. You get price competition, prices come down. Hopefully, we will also continue to get earlier access for patients. So first line or second line therapy, we've seen in both Carvykti and Yescarta get second line indication approval recently. That will continue. That should open up access. But one of the challenges with getting early line access is we just can't make enough products. So it actually doesn't do any good. It doesn't open up access. It just denies even more patients, unfortunately, access to these therapies because they are eligible, but they can't get it. So that kind of efficiency gain and a move to decentralized manufacturing potentially opens up widespread patient access for patients like Opie, who you met, and also, of course, Emily, who pioneered this field for us. So I'm bang on 20 minutes. Maybe time for one question if Josephine doesn't shout at me. Please.
You made a justification of focusing in the manufacturing process in the middle, right? The transduction. In your analysis, if you had not done that and done other process improvements such as batch record reduction, which in my opinion still is one of the bigger timeframes, right? And some other automation, maybe on the QC testing side, what kind of savings would you achieve if you had just done that and not the improvement in process?
You'd achieved zero, and I'll tell you why. Because the constraint isn't QC, it's manufacturing. You can't make enough products. And once we start making products at scale, then that becomes the constraint and that becomes the cost driver. And that, I think it's key for us all to understand, addressing the core bottlenecks first and the others second.
So I have another question.
Josephine's gonna yell at you now, not me.
Why are we still following a very traditional route with BSCs within clean rooms and having a fixed space capacity with maybe 10 or 20 clean rooms per facility? We still are holding tight to that concept. Why?
Should we make that the first question for the panel, Josephine?
Yes. Thank you, Jason. Yeah, appreciate it.
Okay, son. Okay, great. Good. So excited to have this panel. I think we heard some really interesting talks. We had some interesting questions. We'll just address in a moment here. But should be a great panel. We have two Jasons here. So I guess we'll do Jason Ori, Jason Bayer. We also have two Dave Smiths in the room, but the other Dave Smith is not on the panel. So that's good. Okay, so maybe we'll start with the last question here. I mean, maybe I can just comment. I mean, as I alluded in my talk, our main site does have a ballroom as a main manufacturing space, and I do believe that's the path forward. But it also requires equipment or the process to be mostly closed, right? So you might have a couple of open manipulation steps, but they're on the margins, and you have in and out from a grade C to a grade B to a grade A. Yeah, so who wants to start with addressing sort of not only what configuration of sites do we need, but also what clean room grade are we shooting for, right? Is it A, B, C, D, non-qualified, etc.? Yes, that's a good idea. That's a pretty good idea. So maybe you can do first quick introductions. That's a good idea. So we'll start maybe from Jason Bayer.
Yeah, so hi, Jason Dowd from Bayer. I'm the Global Cell Therapy Platform Process Lead. My focus mostly is on the allogeneic side. So there's three auto guys to my left. But happy to be here and can answer those pieces on what we're doing with the Blue Rock assets.
David Smith, VP of Development at BioCentriq. He has 15-ish years of playing around in manufacturing, I think mostly on Autologous as Jason Bayer, which sounds really weird. Jason Dowd mentioned. But obviously a little touch on Allo as that market starts to pick up.
I'm Jason Foster. You just heard me talk for 20 minutes. I'm the CEO of Ori Biotech. We are launching our cell therapy manufacturing platform in a couple of weeks at ISCT.
Hello, everyone. Fabian Gallinghaus, co-founder and CEO of Solaris, the world's first integrated development and manufacturing organization. We've invented, designed, built, and tested advanced manufacturing technologies for both the automation of cell therapy manufacturing as well as the automation of quality control at really high throughput. And we're now deploying and operating these technologies in IDMO smart factories in the US and very soon also in Europe and Japan. You may have seen the recent announcement, $380 million deal with Bristol Myers Squibb for the automation and manufacturing of several cell therapies in their portfolio, both at clinical and commercial scale.
Great. So I just gave a talk. I think I'm good. So let's start the round about prior question. Maybe with yeah, whoever. Yeah, thanks.
Yeah, so for allogeneic, certainly the current designs in the facility that I showed earlier is a fairly small grade B for those open parts of the process. Much probably a 2x larger size for the grade C, and then the rest is I'm trying to lower it as much as possible. Still, these are fairly big, but they're meant to be the design, the HVAC, such that if we needed, and if we had a partner that came in and had more partners, had more open processing, that those grade Cs could go to grade B if needed. But if you're talking about what the future holds, I'm seeing some very interesting designs where it goes to unclassified. Certainly if we can get to the bioreactors, I mentioned that in my talk of where we are with allogeneic, we think that the bioreactors, these are all not grade B suites. So that's where we're headed.
I guess coming more from the allo side of things, lowering CNC, I think 10 years ago, I was tasked with producing a fully automated piece of equipment that could operate in a parking lot for stem cell transplants. And so that's one of the easiest processes, minimally manipulated, hook up a mobilized apheresis, volume reduce it, put some cryo in and freeze it. And could we do that in a parking lot? And I think that has to be the drive. I'm not saying we should get to a parking lot and it's just we go there, but I think there should be a drive to get there. I think, one of the crucial part of why aren't we there and why haven't we got there? A lot in my mind comes down to small volumes. We are terrible at manipulating small volumes. So when viral vector comes in a 200 microliter glass vial that you've got to spike with a needle to get out, automatically you're straight back into a safety cabinet. And so I think, as actually one of my old bosses says, you can't be partially pregnant. So if there's any open step anywhere along that process, you're automatically back into a BSC or an isolator. And that's what's crippling us, is the small volumes, even taking samples, things like that. Anytime we have to manipulate a small volume, largely we're going back into a safety cabinet.
From my perspective, we've built the Ori platform to be able to operate in a CNC environment. We operate it today in our labs, which are ungraded. It's a closed system, so we should be able to make a case to be in C out of the gate, and then D hopefully, and on down. I think the historical reference point is we inherit academic processes that are highly manual, and therefore we have to be in. There's a lot of open steps, and we have to be in B to have those steps completed in a hood. So that's kind of, I think, the historical reference point. And then to your point, I think the underlying point of your question is, why are we, the definition of insanity, why do we keep doing the same thing over and over again, expect a different result? We shouldn't. We're trying to optimize for scale. And if we're trying to optimize for 5,000, 10,000, 30,000, 100,000 products, we need to rethink our current practices.
I'll start by saying why. Why do we even want to go get out of clean rooms? It comes down to cost savings, right? So what we're doing in our commercial scale smart factory in Bridgewater, New Jersey, is we're deploying our flagship technology, the cell shuttle. Cell shuttles have allowed us to reduce the amount of facility size, the amount of facility space that we need by a factor of 10. Compared with conventional CDMOs. So that, to put some numbers to that, we can produce as many cell therapies out of our facility there with 120,000 square feet as conventional CDMOs can produce out of 1.2 million square feet. So that's a 10x reduction in the amount of facility space required. Now let's look within the facility. We deploy cell shuttles in controlled non-classified environments because cell shuttles themselves maintain clean room environments internally, and the cartridges that go into the cell shuttle are closed system. So 90% of our manufacturing floor space is controlled non-classified. We still do need clean rooms to handle the ins and outs. So that's where we're loading reagents into our automation-friendly bottle formats that then goes into cell shuttles. For clinical scale, we're doing it fairly manually, same as everyone else. For commercial scale, once we're getting into large numbers, we're automating, rather outsourcing the automation of that filling step as well, where Gibco cell culture medium is filled into our automation-friendly bottle format by a service provider. And then what we receive are filled bottles with all the reagents that are required for manufacturing process. And then we don't have to do any manual operations anymore because we can just take those bottles, those reagents delivered in the automation-friendly bottle format, put them directly into a cell shuttle and commence the manufacturing process.
Great. That's interesting perspectives, and certainly, yeah, I think we need to drive towards lower classifications, maybe no classification, but obviously there are a lot of hurdles there from a quality and regulatory standpoint. But I want to go back and touch again on one of the points which was made, which is a lot of these processes are coming out of clinical academic centers. They're very manual. Maybe they're already in the clinic with those manufacturing processes. Some of the technological solutions that we've heard about, what's the approach to implementing them early enough so that they get adopted, so that later on there doesn't need to be a wholesale change of a manufacturing process, and that triggers obviously comparability and other activities.
Who do you want to go first?
I'll take it. So when we're working with our early stage biotech or pharma clients, we can take their manual cell manufacturing process, automate and translate it onto the cell shuttle. This is done by our process development teams in close collaboration with the client in about 6 to 9 months. In addition to that, we're offering a standardized cell therapy manufacturing process. So we're offering our clients to leverage the standardized process templates from the cell shuttle. We can easily tune and tweak those processes to the needs of the customer and software. And this allows super early stage clients who may not have done all the process development for their process yet, but they may have a construct, a viral vector, a target, and or some CRISPR reagents to take that target and a rough idea. It basically allows them to take their vector, literally drop it into our standardized cell therapy manufacturing process, give us some direction for how they want us to tune and tweak the process, and we're off to the races. That eliminates a lot of process development for early stage clients and ultimately accelerates their path to IND by several months. It's also got a tremendous benefit for, I think there's some FDA folks in the room. What we're doing here, and ultimately this is part of the company's mission, is to standardize the cell therapy manufacturing process in order to streamline the amount of regulatory overhead that's there. So much process development work is redone over and over and over again in different labs all over the world, and then the FDA is tasked with the burden of reviewing all of these different manufacturing processes that are different enough to require a dedicated review. And we're standardizing that so we can get through that whole process faster and easier, both for the biotech clients as well as for the regulators.
The way we're addressing that challenge is really to empower the scientific community with the technology. We've made a flexible and scalable platform, so you can use it to do R&D, discovery, process development in a lab with a kind of N of 1 on the same instrument with the same consumables, running the same process that you're going to do at commercial scale. So one platform for R&D all the way through GMP. A lot of what we haven't talked about today, which is kind of the elephant in the room, is comparability, right? So I can do a scale down model, I can create it in a bag, but I can't do it at scale. I can do it for 10 patients, I can't do it for 10,000. Using the same platform to develop your process on that you're going to use for clinical manufacturing and ultimately commercial scale manufacturing is a way to get around the challenges with comparability. It's a way to shorten time to clinic and time to commercial. We think we can shave 3 years off the drug development timeline for a product and get it to patients sooner using the same platform at that kind of R&D scale as well as commercial scale. So for us, it's really about providing a flexible platform to empower the scientific community, the academic community, and early stage biotech companies that are driving the innovation so that they can seamlessly transition through the process using the same technology.
I think I might take a slight different spin on it and say that maybe you don't need to automate from the beginning. Maybe there isn't a need to automate. I think if you look at you know, you're probably looking at a 5, 7, 8 years from kind of discovery through to commercial. So do you need a commercial process at the beginning, or are you still needing something flexible, variable, you're learning, you know, that first patient material, you know, it probably looks very different to a healthy volunteer donor material. You're going to have to change your process. So if you've locked everything down, you're in trouble immediately. So yes, automate if you can, if the technology is there, and giving you the flexibility to move forward. I think I joined BioCentriq, and one of the main reasons was noticing a gap for that early stage. Right? There's a lot of great technology out there, and a lot of it is focused on commercial manufacturing. If I'm only doing 5 or 6 times and I need those proof points in order to get my next round of funding or something, I'm going to probably take a very different viewpoint. I'm going to say, commercially, I've got 7 years now. Whilst I'm doing my clinical trials, can I start evaluating the technology? And guarantee in 5 to 7 years, the technology landscape is going to look very different to what it looks today. So I think there is an element of doing too much too early. Now, I 100% agree that we need a plan to stick to, but you also need a plan that you can pivot around in order to do what's right for your therapy. And I think it's a different answer depending on what your therapeutic is, what candidates you're going after and things. There isn't a one size fits all here.
I'll jump right in there, make this a little bit more interactive, because I completely disagree with all 3 points that you made. We've got patients dying on the wait list today, and the science can be as great as it wants. If you can't deliver it to patient, you've got a problem. So at this point, every investor out there, every board member has caught on to this problem. And if you're an early stage biotech, don't believe for a second that this is tomorrow's problem. Your board members are going to push you hard. And they're going to ask you, what's your manufacturing strategy? How are you going to get this cell therapy to patients once it's FDA approved? And if it's FDA approved and you're not on a scalable platform, it's too late. So yeah. I think we need to automate, absolutely, and we need to do it now. Your second point was flexibility. There's this misconception that once you're on an automated platform, the process is locked in. We've actually built an incredibly flexible platform where every one of our customers uses the same cartridge, but the customization problem has been moved from hardware into software. So we can change a manufacturing process at the click of a button. And that's what we're doing in order to support 90% of cell therapy processes out there. And it also allows us to very quickly modify the processes for our customers as we go along, and we go from preclinical to phase 1, phase 2.
We can go backwards and forwards here. But if you needed like a 3-liter culture volume, you guys can't do that. And so there's that type of flexibility. Right? The system you're using is counterflow centrifugation for washing. What if that doesn't work for your cell type? You've got to use something else. So that's the type of flexibility that we have to think about. And we have to work out, is that technology the right one? And I think your technology is very much, I would agree, on the commercial side is what's needed. But
we actually can do 3-liter cultures. So that's also a misconception, but we can take that offline.
Yeah, let's.
Okay, so now for something completely different. On the allogeneic side, so what did we learn from our interactions with BlueRock? They, as I mentioned in my talk, they produced the phase 2 material in Toronto, and we're doing the phase 3 in Berkeley. I'll be honest, that was probably a mistake. In future programs, BlueRock will make phase 1 material and then transfer it to us for phase 2 and beyond. Is that perfect? Probably not, but it's better than what we have, the situation that we have now. We'll solve the problems. We'll generate the data. We'll get the comparability and all the rest. And so, some part of the question was, how do you deal with academics? And the academics are coming to us with, hey, we're UCLA. Hey, we're UC Berkeley. We've got a GMP site. We think we can make phase 1 material. Woof, we made phase 1 material. That was a lot of work. There's no chance that we can make phase 2. And so that's for as a therapeutic developer, we are looking for assets that come post phase 1 because, you need that data. And, Bayer, as you know, is also an investor. So some of the comments here are very germane. But the key there is to have a platform, and as in my talk, I want the same robot. It can handle a T-flask or it can handle a CS10, and or even go up beyond that CS40. But the point is that we will learn a lot by doing the process characterization that we're embarking on. And that 2-year journey starts now with our current asset. And with that, we will qualify our scale downs so that we can do the small scale process. And that'll be the entree point for groups that come to us post phase 1 with their hugely manual process, and then with the ideas that we have with our robots for those robots to mimic what the human is doing, capture that. We feel that that'll be a nice platform for allogeneic therapies moving forward. So again, a little bit different here, but maybe I'll let it go back to the gray.
No, this is great. So we heard basically 4 different views, right? So, and I think to just summarize, one is, do everything manual to start with, play around with it. That's what the academics like. That's what the academic core facility is like, right? And then worry about the rest later. The second was, this is I have a technology I can put in PD labs, in academic centers. They can play with it there. The third was, let me worry about it. Give me your what you have. I'll develop the process for you, and I'll make it for you from phase 1 and on. Right? Those are 3 very different approaches. I don't know that one is better than the other, but they're not all. I don't think they're probably all a good fit for everything. Right? So I think we're going to see a lot of nuance. But to that point, maybe you can speak to. And by the way, I think when you talk about investors, at least from what I've seen, yes, investors are trying to think about the manufacturing process and scalability and cost of goods, which is awesome because that wasn't the case 5 years ago. But more importantly, they want you to get to first in human yesterday, because the clinical proof point at the end of the day is more important than the manufacturing process, although both are super important, right? So the thing is, how do you balance those 2 things, right? As you leave academia and try to get into first in human, right? So there's a balance. But to that point, I'm wondering, what is large pharma looking for, right? Because they usually acquire these assets after there's a clinical proof point, and they're looking for different things. But from a automation standpoint, from a manufacturing standpoint, from a cost of goods standpoint, what are they looking for? We've obviously heard, there's Bayer's represented here. There's a recent deal with BMS here. So I'm interested in hearing what the views are on that. Jason or Bayer?
Yeah, maybe just restating what I indicated, like the schools like UCLA, other academic groups are coming to us, and they want to put that feather in their cap that their clinician, who's usually stand out in their field, they can get the funding, and they can do that. They can really gut out that phase 1 manufacturing. But what they realize soon enough is that it's quite hard. And they all want that funding so that because the universities, at least in California, they all want those startups. They want to churn those out because that gets also the investment and the endowments back to the university. So in my presentation, Bayer's an industrialization engine. So we'll take assets post phase 1. We could probably take them much later as well. But, in terms of what an ideal would be is post phase 1. We can then industrialize it, get to that medium level process, and then concern ourselves with the appropriate scale that we need in order to drive those cost of goods. And for an allogeneic application, I don't think about it in terms of cost per dose. I'm thinking of, okay, well, how do I hit 20 billion, 50 billion, 100 billion, and 1 trillion cells, and going on beyond that. And I have broadly, what's my bronze medal, what was just sufficient to get me on the podium, what's silver, and what's gold. And, gold, we have to push the envelope. And there we're looking at trillions of cells per batches, and then, producing multi batches, multi doses, because the goal here again is cost of goods. So I'm looking at it from how many cells that I produce. Hope that helps.
I'll jump in to say, since I'm not a big pharma player, I can sort of be candid about this. What they're looking for is products that can make money, that they can serve a lot of patients with, because they needed to deliver an ROI on the massive investment that we make bringing products to market. It's like $1.9 billion to develop and launch a cell therapy product. I just saw a recent report on it. It's a massive investment that we need to ensure viability of that product in the market. And having products go all the way through the process and launching them only to then fail commercially is a disaster for patients. It's a disaster for investors, and it's a disaster for companies. If it's going to fail, we need to fail early. We need to have a plan for manufacturing early. And that, for me, is where the discussion needs to go. I don't think the discussion is being had enough today. We're still focused on safety and efficacy, talking about safety signals. That's all we care about. Get to the clinic fast, first in man. Give me a signal so I can raise my Series A. You're not going to get partnered, and you're not going to get funded unless you have a plan